Yeah, no, I think you did that last time. You just can't put anything by escape in there.
Yeah, watch anything.
Morning. Not quite. Thank you very much.
West.
Maybe we got a fish.
Maybe we did.
Okay. I raise up that. Come on. Bye.
Okay.
Está bien. Yo quería abeja, venir, pero no sabe. ¿Tú lo puedes hacer? Es que Jessica no quería.
¿Estás permítame?
Sí, sí, parece que
Sí, sí, que se lo puede.
Yo creo que le digo.
Is it West or you guys anywhere?
Yeah,
No,
no,
no
we
me
just
contestó.
moved people now towards where we're going now. Interesting.
Tell me más.
Oh, you're coming into the good.
Tell me.
Yeah, we'll just try and video transect now until waypoint 8.
Okay.
And then,
And then it's me.
yeah, then,
My babies.
yeah. So the only collection sample that's left is number 2. Don't put anything in there that might climb out because it's not properly closed.
So which viable can I use
This
to
one.
use my baby?
This one.
Which is?
1B. 1B.
West is running.
And no more collection of anything else, right?
In your watch? No.
It's only whenever we reach waypoint 8. Jessica, whenever we are now going to waypoint 8, we need to be in transect mode.
1 meter off the ground.
Yes.
Maintain.
The lasers is down.
Bridge, new waypoint.
Updated waypoint.
gonna do transect until waypoint 8. So we are in transect mode
Yeah.
from now onwards.
Okay.
In transect 8, then we sample. My babies again.
The coral babies or the Holothurian babies?
Barrowby Bridge.
Yeah, bridge.
I'm gonna have to just come a little more straight ahead to dodge a bit of ice.
Roger.
Maybe a little bit from my brain, so the shift can dodge zones. So if they come up with it, I'll just deviate and I'll be a little bit that way for now.
So we still have the fish in that. One that we saw actually came out from around the push post. So fingers crossed it makes it to the surface.
What is it?
That brown is just not the same as the thing.
Communicate and then you do it. I'm sorry? I will now say something.
Okay.
Then you do it. Yeah? Olá, muito boa noite a todos. Bem, estou aqui de novo a tentar comunicar o que é que nós vamos fazer. Neste momento nós estamos a fazer um transect. Vamos estar assim um pouco porque temos que estimar as densidades dos organismos que existem aqui, por isso não vamos a mostrar nada e estaremos durante uns 15 minutos mais ou menos, pronto, só num transeito. Neste momento o que vocês podem ver é que o sedimento continua a ser composto por lama, tem holotúrias, tem crinoides, tem também ouriços, tem poliquetas, que são aqueles tubinhos que vocês podem ver no sedimento. E para já é isso. Mais à frente estamos a ver anémonas. O meu nome é Teresa Amaro, eu sou da Universidade de Aveiro, Portugal. Estou neste cruzeiro ou nesta campanha oceanográfica porque estou bastante interessada em estudar as cadeias tróficas, ou seja, estou a tentar descrever como é que o ecossistema nas mais variadas estações aqui na Antártica está a funcionar. Para isso e como indicadores eu recolho sedimento, recolho também holotúrias, os mais, ou chamado nome comum pepino do mar, e também recolho água e corais, isto porque os corais filtram a água, alimentam-se assim, são que nós chamamos de filtradores, e as holotúrias ou pepino do mar comem o sedimento. E a partir daí, quando faço análises, eu na realidade consigo dizer como é que o ecossistema funciona. Uma poliqueta é um verme, uma minhoca, que existe no fundo do mar. Se vocês conseguirem ver uns tubinhos que estão a sair do sedimento, esses tubinhos está uma minhoquinha e é isso que nós chamamos de uma poliqueta. e obrigada desde já por estarem super interessados em seguir naquilo que nós estamos a fazer aqui. Vou passar agora à minha colega Amber, que também se vai apresentar.
Good night or good afternoon or good morning everybody. Here right now at Falkor it's shortly after midnight. We just took over our shift in the rough dive 780. We are in front of the Bach ice shelf and we are basically surrounded by a lot of ice. And yeah, we're doing this cruise called Climate Connections at the Ice Sea Interface cruise. Is what I think is the 6th dive of that cruise, and we already are quite full. The rough is quite full. We already did all the push cores. We sampled a lot of corals already and sucked up a lot of stuff with the suction sampler. There's some things in the bio boxes. We still have a little bit of space left. We also have a little bit of more hours of dive left, and right now we're transiting from waypoint 7 to waypoint 8. We're almost halfway there to see and characterize what is here on the seafloor. We are at a depth of 740 meters. And yeah, have fun watching the live stream. Yeah, my name is Amber Henningsen. I'm from the Institute Senckenberg am Meer, which is in the beautiful northern part of Germany. And I'm mostly studying macrofauna, especially isopods. But on board we're sampling meiofauna, which is even a smaller size of animals that you, in the normal case, are not able to see with your eye. That's why we take a push course, and we're gonna analyze our samples back at home in Germany.
Nice.
Yeah, so I saw a question in the chat, how did I get into this line of work? So it started 2.5 years ago. I always wanted to go on a cruise. So always wanted to participate working on a vessel in the field of science. I originally studied geography, so I have some insights in that already, with mostly paleoceanography and mostly bathymetry and mapping the ocean floor. So I started on a research vessel, Ronne, which is the biggest German research vessel. And then I was there and did more or less an internship, and I got another opportunity because I met someone on board that is now my boss. And I don't know, I think he was just happy with the way I worked, and I kind of got into the biology field and
Nice.
receiving sponge.
Can you zoom in? You okay?
So yeah, it was more or less a very lucky accident, but I'm of course super aware that I'm super lucky to be part of this expedition.
Very nice. I'm going to introduce. So the shift is composed by myself, Teresa, Amber from Germany, and then Sveta from Russia, and Benjamin from Chile. Sveta, do you want to say something?
Sure. Go ahead, here's the mic.
Thank you. It's nice, really cool. Sveta, there was some Russian people in the shots, so maybe introduce yourself in Russian.
Hi everyone, and всем добрый день, здравствуйте, если кто-нибудь слушает по-русски. So I'm a paleoceanographer and a geochemist, and I'm a research fellow at the University of Cambridge and the British Antarctic Survey from the UK. So I study basically sediment cores and specifically these small shells in the sediment cores. It's very interesting in Antarctica, it's a very understudied area, so we've been getting really good sediments so far. We've checked some out under the microscope, there's some very good specimens in there, so hoping to improve our understanding of past ocean climate change. And essentially that data can be used as a natural experiment for the numerical climate model simulations, which then will help us improve future predictions for stuff like sea level rise, specifically from Antarctica, also CO2 increases and temperature changes.
Jessica, can you just zoom in here? I think it's a nice sea cucumber. Benja, can you say something? Só un poquito en español. Solo para presentar un poco.
Hola a todos, buenas noches. Les habla Benjamín Aguirre, estudiante de Biología Marina de la Universidad de Valparaíso. Bueno, en estas instancias nos encontramos en el mar de Bellingshausen, en la Antártica, explorando las profundidades del océano. Como pueden ver acá en la pantalla, hay un par de anémonas y unas cuantas ophiuras. Es muy interesante toda la vida de las profundidades marinas y gracias a este ROV que tiene esta embarcación del Falcor 2 podemos verlo ahora en vivo y en directo.
Thank you, Benja. Maybe I should also introduce myself and try to say also my career path. So I'm Portuguese. I did my PhD in the Netherlands. I always wanted to be a marine biologist, so I left my country and some time ago I went to the Netherlands doing my PhD. After that I went to the UK in England, in Southampton, where I actually did one postdoc of 6 years, and I actually started to do deep sea research there because in the Netherlands my PhD was not really in the deep sea. After that I went to Italy, to Greece, to Japan. Actually I went to several countries, always looking and interested in really interesting things. At the moment I am back in Portugal. I actually do something more than deep sea research. I have a project in Cabo Verde, which is in Africa, in front of Senegal, where I do mesophotic actually research. So mesophotic is everything that is beyond 30 meters. So for that I had to apply what I'm actually doing in the deep sea into the mesophotic environment, but instead of using an ROV, I am the ROV. So I use rib reader and scooters and I collect my own samples. So it's actually quite cool, and it was very interesting to do all this deep sea research, from my past, because it helped me to apply what I learned and then do something completely different. Now in the Antarctic I'm back to the deep sea, and I am actually quite excited to be here and do deep sea work using the ROV.
What would you like to sample today?
I already said. But se calhar agora em português, porque temos alguns ouvintes que estão a nos ouvir do Brasil, Portugal, até mesmo da África. Nós tivemos-nos a apresentar. Está aqui a minha colega Amber, que é da Alemanha, a Sveta da Rússia e o Benjamin do Chile. Eu própria sou portuguesa, como já vos tinha dito no início, sou uma ecóloga e eu tive um longo caminho lá, desde que fiz o meu doutoramento até chegar a este momento. Esta campanha oceanográfica está a ser realmente bastante interessante. Somos investigadores de vários países, multidisciplinares, estão biólogos, ecólogos, geólogos, oceanógrafos.
Yeah, sure.
Alright, I'll give it a try, and worst case I'll have to move a little bit to dodge that ice again.
Tivemos que nos desviar do nosso trajeto porque havia um iceberg e agora, já que estamos de volta na trajetória que nós tínhamos planeado fazer. Quando chegarmos ao waypoint 8, iremos a mostrar pepinos do mar para a minha investigação. Oh, nice. Could you please zoom in? Thank you. Thank you. Wow. Really nice. Thanks. Well, to answer to the question, well, I'm not really sure. At the moment we are at 740 meters and our waypoints are a bit shallower than this, so I think we are going up. I'm not sure if we're going to see a rock, but possibly yes. So let's see. Whenever we arrive, we need to start some. Yeah. Thank you. Yes, please. Yeah. Very nice. Wow, that's so nice. Is it possible?
We're just going to look for ice right now.
Not sure how the situation is because of the iceberg. Is it okay?
Yeah, so it's just moving a little bit south right now.
Yeah. Oh, really nice, Ben.
fish
Really
there.
nice. Sorry?
Some nice fish.
Yeah, and sea pen. 2 sea pens. Nice. They're not.
No, we're not docking at all. We're at the more or less. I mean, I would not call it open ocean because there's ice around us, but of course here it's so remote that there's not that I know of any port. So if you would want to go on land, then you would have to put your anchor out and find a way to make it there. But there's not even a station close to where we are right now, so we're basically sailing all the time.
Another sea cucumber. Nice.
Do you collect some already on this?
Yeah, but I will be collecting in a while. Because I need to compare between depths. And so this dive, I started more or less around 1,000 meters. And it's planned to finish around 600. And because I'm interested in looking at how the ecosystem works, I like to compare these different depths. So I have collected sea cucumbers at 1,000 meters. And whenever we are reaching 600 or about 600 meters, I will be collecting others so I can compare and see if the ecosystem is functioning in the same way.
So I think Teresa in the room is a professor of sea cucumber. She's a
Not-
big expert. She is. Compares them to the ecosystem around. It's just very interesting.
So once you reach the point blank, would you like a sediment sample or?
Not yet, not yet. No, we are not doing any push holes. For the moment we're going to be looking, but I'd like to have it a bit more shallow. And we are going to actually collect sea cucumbers, and then it's only then transect them. It's very easy.
Yeah. Relax.
It's okay if I won't speak.
Teresa, what is your favorite sea cucumber?
What?
What is your favorite sea cucumber?
Scotoplanis. It's Scotoplanis globosa. I will point it out whenever I see one. The common name is sea pig. They are really nice to sample, and they are actually cute to me.
So Sweta, what is your favorite foraminifera?
Sibosaudes willistorfi.
Okay. Can you point that out if you see it on the dive stream?
I'm afraid foraminifera are very small, like grains of sand, so we have to get the sediment first and then look through the microscope.
Did you already see it on this cruise?
Yes, we found some.
So we're just about midway.
Mhm. Just please. Yeah. Nice.
Amber. Do we know what the little worm tubes are? Why are they here? There's quite a few.
Should be polychaetes. I mean the tubes of polychaetes.
I already said that.
But further than that, I don't really. You know, Tony, do you know what they're called?
The tubes
Yeah, the tube
could
worms.
be serpulides,
Ah, okay, Serpulides.
tube worms.
Yeah, sometimes when you see them, then they are kind of branching.
You can see the tentacles looking out of the tube.
So replying to some questions that are in the chat. During my shifts, I haven't seen any plastic. Have you guys? Have you seen plastic?
Not really, but
You can see that we still have a little bigger bogey bit just in our port bow, and I'll pull back and go to port side to get that past the bow first. But it's so close now that it probably doesn't affect you, me trying to hop around it.
So good.
So on the last cruise, when we were in Chile, in the Chilean margin, there was actually someone working with plastic. And we saw a couple of things. For example, like the stick of a broom, a plastic one. Then we saw multiple trash bags, which was funny because every time we saw trash, there was one time where we saw a newspaper folded open. It was from the year 2021. And there were sea urchins that were reading that page really closely because they all accumulated on that newspaper page. That was actually funny. I mean, there was some. I would say here in Antarctica is less.
I think we haven't seen any big plastics, right?
No.
Well, to be honest, I'd like to collect sea cucumbers where, more or less in the area where the push cores are going to be taken. So that's why I want to be a bit shallower.
But the remaining push cores are only the one from
No, they are already collected.
Okay.
But after this dive, we're going to have another one only for push cores. And so if I have the sea cucumber more or less at that depth, that's good. That's fine.
I see a nice rock.
You want to sample it?
There's a brachiopod.
Yeah.
I do like nice brachiopods. Do we have time to sample some brachiopods?
You wanted those two?
I'd have some brachiopods, please.
Jessica, do you think it's okay if you sample these little guys here for Sveta?
But they cannot go with the other brachiopods, right?
They can go all together with all the
With the ones that you already sampled?
Yeah, all the brachiopods.
Because
So
then
know
you
where
would
they are?
not lose another suction
Yeah.
thing. I think you in
Okay.
3.
So 3. And if we can go to number 6.
Negative. I have to pull back a little bit more, go south, and then I can go ahead.
Rachel, we'll just follow your lead for this moment. And when you're clear, you just tell us.
Yeah, I don't think it's going to take a whole lot longer.
Let us know. Yeah. Sveta, I'm sorry. We cannot collect because
we have
there's
to
an
leave
iceberg. Yeah. We have
next
to-
time.
Yeah. So at the moment we cannot really sample because we are running away from an iceberg. So Sveta cannot have her brachiopods.
So I'll explain the brachiopods. I only want the shell and not the living thing, so we're going to share, and Teresa is going to have the organism, and I just get the dead shell. So my geological heart likes shells and fossils. But the reason they're so interesting is because in Antarctica these brachiopods take a while to grow, maybe up to 400 years sometimes, and essentially they build their shell layer by layer. So what you get if you study chemistry of these bands, you can reconstruct climate back to a few hundred years ago, depending on how long the shells have been alive for, and have a really high resolution record of, for example, things like past ocean temperature and salinity changes, which might help us to understand the sort of anthropogenic effects on the ice shelves melting in this area.
Thanks, Veta. And thanks for sharing the animal with me.
Sharing is caring.
I've said earlier that I'm actually interested in how to know how the ecosystem works and functions. And for that, normally I would be sampling
Hey Claude, you're
every
right.
kind of
I'll just go
animal.
try and pause, see what happens.
And actually use isotopes.
Continue with the transect. I'll be good. We already moved to the location that we wanted to do this sample.
Okay, in that case then I have to keep moving astern and to port side a little bit longer to get the ice on the other side of the bow.
Roger that. Just let us know when you're all clear.
Thank you. Nice.
This is very nice view.
Yeah. Also that they move their tentacles or whatever in differing
Yeah,
rhythms.
it's really cool.
Yeah. Someone likes emojis.
So someone in the chat is commenting about cool urchin hairstyles, and actually earlier today we saw something that I thought looked like would make a nice wig. It looked quite fluffy.
But you also got a new haircut, huh?
I did get a haircut today.
You're not French now, do you?
Oh, and you didn't see the second half of the
Oh,
haircut.
I saw.
Oh la la.
Was it Rachel? Or you did it yourself?
No, I had Professor Tom and myself.
Oh, yeah.
As I cut Professor Tom's hair the other night, everyone else is welcome to the hairdresser. It's open 2 a.m. until 5 o'clock in the morning every night on the
It's
deck.
a very inconvenient time for a hairdresser.
I think it's quite good. It's nice and fresh on the deck. The hair just flies off the ship. You don't have to worry about the drains.
True. Maybe the stuff that we saw earlier was the remnants of Tom's hair.
Maybe.
Probably.
Did look quite blonde, to be fair.
So as you guys are listening, we actually do more than actually science here on board. And today, or in the morning, after I actually process the samples that the ROV is going to bring for me, I'm going to do something really, really nice. So I will be connecting to a school in Portugal, in Porto, from kids of 10 years old. And I'm going to try to show them how the ship is, what we do, what an ROV is. And I am going to try to answer to all the questions they might have. So it's quite interesting thing to do. And I'm going to try to actually introduce them to the different jobs that they can have in the future. So it's not only about science, but all kinds of jobs that they can have towards this scientific actually expedition. So I think it's really nice that the kids of 10 years old has a spectrum of actually they can be. It's not only teachers in high school or engineers or lawyers, economists, they can do a bunch of other things. So I'm really looking forward to show them this, although I'm gonna be really tired after my shift. Sea cucumbers. So we are deviating again, right?
Yeah, I think there's it's just still like a little bit of ice.
But then we're gonna try to go back.
So I think for the viewers, I thought it was very cool that Teresa, when she gets her sea cucumbers, she dissects them like a surgery. We have a surgery set up in a really cool lab with this kind of red lighting. It's quite semi-dark in there, and it's very cold, like a fridge. And then she
It'll be done in
dissects.
another 3 to 5 minutes.
Okay.
Copy that.
Dissects the sea cucumber and takes out some bits from the inside.
Guts.
And it's really, I think it's really interesting.
Did you watch that?
Yeah, I tried to watch a little bit, but I also get my samples at the same time, so unfortunately you can't watch the whole thing happening at the same time. Have you seen it, Amber?
No, I haven't.
Because you also get your samples, right? So you
I also
have
get
to
my
process.
samples, and I'm quite light-hearted when it comes to organs and blood, to be honest.
But it doesn't look like organs in this thing. It kind of, like, it looks a bit
Like
like
a
a
jelly?
big jellyfish, and then the thing she pulls out is kind of like a darker jelly, so yeah.
And maybe I will give it a...
I think just come in when the cucumbers are on the surgery table, or maybe should we say pathologist?
Yeah, that's better probably. I
We'll
think
tell-
Teresa has not a good survival rate as a doctor.
Oh, that's true. So maybe a pathologist would be more appropriate. Teresa, you have been labelled the pathologist.
I cut everything I see. I brought my dissection kit, so
It's perfect.
be aware.
Harvey Bridge, I think I can start moving ahead, but not to starboard at the moment. Would that work for you?
Yeah, Roger, we have decided that we're going to go to waypoint number 10.
Yeah, I can go in that direction, no problem.
Okay.
Perfect.
Just put a waypoint. And you could deviate to suit.
So I'm gonna now translate what I said in Portuguese. Depois deste turno, quando o ROV estiver à superfície e trouxer todas as amostras com que eu tenho que trabalhar, vai haver uma transmissão live para uma escola em Portugal, no Porto, onde vão estar miúdos de 10 anos e nós vamos lhe dar uma tour aqui no navio. Vou-lhes
Going
mostrar.
towards south of number 10.
Roger.
Vou-lhes mostrar as diferentes profissões que podem ter, porque eu acho que é bastante importante mostrar a uma criança diferentes trabalhos, não aqueles trabalhos mais comuns, como ser advogado, economista, professores, mas que podem ter um outro tipo de trabalho. Então eu acho bastante importante, na idade que eles têm, dar-nos uma transmissão ou uma lição ao vivo daquilo que pode ou existe no mundo. Estou realmente desejosa por esse momento, porque acho que vai ser muito entusiasmante. E sei que miúdos dessas idades têm bastantes perguntas, e perguntas até às vezes bastante difíceis de responder.
It's gonna take a while. When you're almost at the height of waypoint now, shall we try to fulfill that for another minute? Okay. Do you see some? That was the reason before. Yeah, because Sophia put it in. Anyway, maybe it's not the most updated one, is it? So, was there multiple versions of 780? Yeah, because I went after the science meeting. Okay. We changed it.
Maybe she just uploaded it into the Google Drive or something. You can check.
I don't know, but yes, let me know. Yes, we have, of course, people dedicated to network and software issues. It's a really, really nice ship. Oh, sea cucumber, really cool. It's really different, huh? From 1,000 meters.
There's another sea cucumber.
There's not.
Yeah.
Octopus.
We are at about 770 meters, and if I see the composition of the megafauna, meaning the animals, the organisms that are, that you can see with your eye. Oh, Sveta, there is a rock.
Oh, rock.
Oh, wow. And really nice animals.
Maybe I should get a t-shirt that says I love rocks.
We need to collect actually a rock, but not yet.
Not, it's too big, I think.
Yeah, it's very big.
What is that mushroom?
That one on the left?
Yes.
Anemone.
It's an anemone.
Oh.
So we are trying to convert Sveta here into a biologist because she only loves rocks, but now she's learning all the different animals that appear in the deep sea.
I think I'm very good at identifying sea cucumbers now.
Oh yes. And at the moment we see, coming back to sea cucumbers, you see 3 different species. Species from the Elpidiids family, but also a different one that I actually don't know the family name, but it's really big and very similar to the sea cucumbers that we can see in shallow water. And whenever you analyze them or you dissect them and you analyze the tissue, you see that these guys, they move in slower motion than the other ones. So the other ones are more competitive than these ones that move really slowly. And so the other guys, they actually eat the good quality of food, whereas these ones don't. Yeah, Sveta, you learned the lesson?
I did, thank you, I'm taking notes.
You have to give us a class on rocks.
I'll have to wait till we see some.
Because for me, a rock is a rock.
Well, the rocks can be very interesting. Essentially, they're like products of the earth. So then you have these sometimes like spots on the rocks, which actually tell you surprisingly a lot of the kind of like eruption that it came from, the origin of the rock. So you can have sedimentary versus kind of metamorphic rocks, for example. The metamorphic ones come from plates and tectonics and earthquakes, if you like, when the rocks are kind of burst out of the soil, whilst the sedimentary rocks is when sediment accumulates, and over sort of billions of years of time scales, eventually it builds up and makes these rocks, for example, limestone, which we use for really beautiful architecture, for example, in Cambridge or Bath or in Exeter in the UK. And if you look at the limestones through the microscope, you actually see that it, again, it's made up out of these calcium carbonate marine organisms, the one I'm interested in, and some other families. And essentially any rock is like a beautiful archive of the past.
Very good, Sveta. Thank you so much for the explanation. Huh?
I think she has a point.
Yeah, of course she has. Oh, another interesting sea cucumber. I want to collect those ones.
This brown one? Sausage looking like?
Yes.
Quite different from
What
the
is this
pinky
red
ones.
thing flying by? Is that
Jelly.
jellyfish?
Mhm. Oh,
Oh, look at that fish.
can you? Yeah, to me. No. Maybe clean that up anymore. It's nice. Yeah. Oh wow, did they? We need Tom. in the room.
It
Oh,
looks a
Tom.
bit dead, huh?
Tommy,
Tom, he's commenting. He said
it's
it's a blunderfish.
blunderfish.
Okay.
Ah,
It's missing a few scales on the back. At least it looks like.
really nice.
He said there's a little layer on the bottom lip which helps us to identify the fish. Thank you, Professor. Mr. Professor Fisherman.
It says the fish has his haircut.
Yeah.
Really nice. Can you zoom in here, please?
Maybe that was the inspiration for Tom's haircut.
Probably. Is that a coral?
Oh, nice. Could you zoom in and so that we can take a picture? Wow, nice.
Ooh.
Marita, you have that one already, no? Or you?
I think it's the same that
Yeah.
we collect.
I see too.
Nice, thank you. As I said before, we come from different fields of expertise, and on board we have Tom, Professor Tom, that is a specialist on deep sea fish. And it's really nice to have him here because we are actually learning a lot about fishes, at least I am.
Yeah, me too. They're really cool. He sort of nails them down to a chopping board in the lab and then applies the formalin, which I cannot say that word, the biological, Teresa, how do you call that? Chemical? And then like preserves them so they're going to be museum specimens. And occasionally also dissects some, does some surgery to the fishy.
He's doing the preservation here on board?
Yeah.
Crazy.
It's pretty
Whoa.
cool. They're
I
like
didn't know.
nailed down to the chopping boards in the lab, and then I get a nice fish smell whilst analysing my pool water. So it's nice.
I think it's quite impressive that he is able to preserve the fish.
Being Portuguese, it's a pity that the fish cannot be eaten. Otherwise, we eat. I would have tried to eat the fish.
Apparently, Tom said they're not very tasty.
Yeah, but I don't know. I need to try. Maybe I like it.
I still think we should try some sea cucumber. Any
I-
excess you have.
I tried. I was forced to try actually. On an expedition I had in Japan, we were actually cruising for 6 weeks, and I was actually doing an in situ experiment using sea cucumbers at 5,000 meters. I actually went on a submersible. It was really, really cool. Anyway, my colleague from Japan, whenever we arrived in, land in Japan, he thought it was very funny that I actually should try the animal that I work with. So I was kind of forced to taste a sea cucumber with 2 shots of sake. So to be honest, I can't remember the taste because I drank the shots. And I closed my eye. I closed my eyes because I had a raw egg in the middle of a sea cucumber. But of course I had to please the person that gave me the possibility to go to 5,000 meters, and I did it, but it was hard.
So to answer the question in the chat about rocks, someone's asking if the last 2 rocks that we sailed by were metamorphic. I'll say maybe. Is that? But I think, yeah, perhaps they did look like the sort of the drop stones that the glacier essentially spits out as the ice sheet retreats and melts and the icebergs kind of separate from the main ice shelf. But Teresa, going back to your sea cucumber dietary story, So it's Sasha is who's one of our co-chief scientists.
Is that a rock?
A rock. Quickly document the rock.
Rocka.
Yeah. Wow, it's nice.
So this one looks like a very large. Oh, it's actually like a bit of a terrace. Take a photo.
Yeah.
So maybe we have a bit of a terrace essentially here. So perhaps the stuff on the left is slightly deeper than the stuff on the right. So kind of like rocks, but it's hard to tell from this shot. But yeah, so Sasha, who's one of our chief co-scientists, and he's a professor of rocks. No, he's a professor of the bottom, the topography, so the mapping stuff we do from here,
Really
geophysical data collection. It's his birthday on the 27th, so I think it'd be really funny if we asked the chef to cook him some sea cucumber.
Yeah.
What is the
Let's
stuff
do it.
on the left? Is it the Alerida?
Yeah, I think
Wow.
so.
Look at these beautiful
Wow,
rocks.
so beautiful.
Maybe we should collect this big one for Andres, who is actually a professor of
There's
rocks.
can we zoom in?
Could you please zoom in? Yeah.
Oh, look
Here,
at all these
there.
cute brachiopods.
Wow, beautiful.
It is huge.
Nice.
Are you admiring the rock or the animals?
The animals.
I'm taking good pictures. Thank you, Jess, for the zoom in observation of the
I
rock.
don't know if anyone's interested in that. Probably not.
What
We
is
don't
this
have
white
space.
Probably Nemertin. Nemertin.
The Martinez, probably?
I don't know.
A huge one. Do you
Several.
Think, but do you think Americo
No,
would
no.
want that? Americo?
Americo?
Sample?
Yeah, collect.
Teresa, can we collect the long white worm
Um,
for America?
No, I mean, I don't know if he wants Nemertains. I know he works with polychaetes.
yeah.
Oh.
No, just
polychaetes.
go on, please, yeah. That's very nice, Sveta.
It's
Looking.
very nice rocks. I'm taking screenshots.
Do you want us to collect the rock?
Maybe we should collect a little piece of rock for Andres, actually, because
Yeah.
it does say in the plan, right? That
Yeah.
we need to collect
Yeah.
some rocks for
Yeah, yeah,
Andres.
But
I think on the smaller side of the
Yeah.
rocks, maybe we don't need a massive
we
one.
need to collect a rock.
Rich, can you come to us with pause here? Just gonna collect the rock. Yeah, thanks.
Now it's gonna be interesting. I don't know which rock.
I think we just need a small piece of rock because
Yeah.
these all look quite homogeneous to me, and already rocks are quite heavy items.
Okay, yeah, we can probably sit down there. Maybe some Geological.
What? This one? I don't know. The one that actually is easy for you. Oh, it's very impressive.
Oh, it's a beautiful wall.
Yeah.
Rock wall.
Yeah?
¿Se puede cerrar una esquina aquí?
Aquí? Sí, después de la colección de la...
There's a rocky one.
Yeah, just
A
keeps.
few.
After the collection, we need to close one missing.
Teresa,
Thank you.
Teresa,
Yeah?
if we have some time, could we suck me some brachiopods from this rock?
Yeah? Where are they?
You can see some on the left. I think they're just sticking out, maybe.
Ah, there. Okay.
But if we don't have time, that's fine.
Let's collect the rock. Yeah?
We do have a lot of time.
Oh.
Actually, we have the dive until 5.
Okay.
And we are at waypoint 9.5, so. And Patricia said we should wake her up when we are finished early, and I don't want to wake her up.
Oh yeah, no, let's just
No,
keep
we
going
haven't
then.
finished.
No. She was saying if you notice that we're coming more to an end before
Ah.
5, we should wake her up.
No,
But I think we should just.
I need to collect my babies.
I
Is there-
know.
Oh.
We still have.
Can I
That's a smooth
Yes.
rock action.
one of these here? Those
Yeah?
these little guys.
Yeah, I think the
Yeah.
small ones,
Yes.
those look fine to me.
Yeah. What? Yes. One of those. Tom's fishes are very active here.
Look at that prawn on the left. What is
Yeah.
it? It's not a prawn, is it? It's oh, it is a shrimp.
Yeah.
My biology's improving day by day.
Yeah.
Very good.
Nice. Chew.
Sometimes
Yes,
they're called
I mean-
ceratopsies, but this can also be something else. Just know the stuff from the Pacific.
Really cool.
Oh, nice shot.
Very nice.
We can get a good dark background for the shot.
Mm-hmm.
I guess there is a
Yeah.
Okay. That's good. Which blocks on vacation? Was it 2 or 1?
Yeah, these two, they don't have anything, so, but I need one for the, oh.
Can we put them on the
It's
rock
dropped.
basket?
Yeah, in the rock basket, I think it would be better, yeah. We need these two empty.
Okay. For the rock basket.
Yeah.
Rock for. Exactly.
Not yet.
Fell off.
Yeah.
Oh.
It's here, I think. Yeah. The rock basket.
Even better.
Oh,
Yeah.
beautiful rock. Thank you, Jess.
I'll wait until.
Yeah.
Yeah, so it's cool.
Yeah, it's cooler.
Yeah, it's nice.
It's got some nice red sort of shiny things on it.
I think it's manganese.
I think you're right because we had some from a dive earlier on that had a manganese coating.
Mm-hmm.
This is the reason that this manganese basically sort of builds up on any pebbles and rocks and any other kind of fossilized shell things in the sediments, on top layer of sediments in the deep sea. So just from the kind of reducing lack of oxygen.
Yeah, but I think America is gonna enjoy to look at this rock before you take it, because I can see a polychaete.
Oh, 2 in 1.
Oh yes, two. So. Yeah.
So is that rock basket 1 or 2, Teresa?
Wait a moment. Yeah. It's this one, right? I see. Whenever you have a minute. Is basket one. Yeah. Rock basket 1, I think.
Rock basket 1?
Yeah.
These could be suck me a few brachiopods off these
Yes,
rocks.
yes. Nice.
And you said they can go with the others?
Yes, they can still go together.
So, yeah, we need. Yes. Do you know what that is? We will, sort of, yeah. So here,
White shells.
see, you see a lot of them.
Yeah.
If you can put them in sections and put, well, 3, yeah, because there are others there and they can be together. Right, Sveta?
Yes, yes, they can go together.
3. Yeah.
Some of the property cards, which we will explore placed. And this is going to be.
Yeah.
And then after this, we're on this one.
Yes. Yes, please.
So can you instruct us?
Yes.
Is it on the right side there? or-
Well, here you have, but well, here you have, so it's actually
They look like little circles.
yeah, yeah. You have everywhere, right? Here as well.
Yeah, you can look at that next time.
What are these white flower things next to my brachiopods?
I think sponges.
Yeah. You have sponges and creamers.
Circles. Like circles there. Not just flies here. Do you mind bringing it on the screen?
Yes.
I'll try to make it and I'll turn this section out. Alright, you are all great.
Yeah.
Yeah,
Later.
Oh,
Yeah.
oh, thank you, Jess.
That's really
Nice
nice.
blood.
Oh, yeah, that's
blood
lovely.
good.
It's good. It's good.
Oh,
Thank you.
I love the shells, thank you.
Thank you.
We
Really
got
nice.
this
Yeah.
Yeah, we got a lot. Some
So-
coming in.
Oh, we get some more?
We keep shaking it.
No, it's enough, right?
I think it's loads, thank you.
Zero deception.
Yes. Then Niskin, Maritza.
Where are these going?
Suction sampler number 3.
Number 3.
Okay.
Don't forget to log in.
I am looking at, don't worry.
Yeah.
What is it? So this is
It's a
geology
rock.
sample?
You have a pen?
Yes.
So the Niskin bottle, I think, is number 5, 6,
Suction
I
sample
think.
3.
Yes.
I think it's 5.
Do you want a certain height off the ground for this?
No, here. Yeah, I think next to the rock. It's very important. As close as possible.
Oh, it's a little bit closer to the rock there. Alright, so this is gonna be another bag.
Yes. Number 5, Niskin bottle.
How do I look
We
at
coming?
Oh
or-
yes, I can see it,
Right
sir.
down?
Yes.
Yes. Don't
got
forget.
Okay, so let's go there after.
Nice.
Oh, that is quite smooth action.
Yes.
Someone was asking in the chat, does a big rock create a microenvironment?
Oh yes. Actually, rocks are very nice for biologists because they create a different ecosystem in all kinds of depths. So a rock for me is a rock, but it's very important for biology.
You guys want to keep the other shots of this, or this is it?
Yeah. Yep. Very nice, actually. As you can see in this rock, you have all kinds of organisms. So it's really, really nice.
Is there thing sticking out here? Is that bryozoan or is that coral?
Where?
Like that one?
That is a good
Or maybe it's
question.
just completely something else.
I would say it's a sponge.
Yeah, I don't know.
Okay.
Sponges have the weirdest shapes, so if I don't know, I always think it's a sponge. They come in such weird shapes.
It's a beautiful shot.
Thank you. Oh, look at your brachiopods.
I know. Everywhere.
You have enough.
Okay. Yes, boss.
This dive is actually, or has been, very productive. We have collected all kinds of samples, meaning that whenever the ROV is on deck, everybody's gonna work a lot.
That is going to be at 5 o'clock in the morning in case people are listening in different time zones, so
Yeah.
excellent fun. Teresa, I have a question about the sea urchin.
Oh, yeah. Okay.
So you know it's like looking nice and pink, but there's a few of them.
Where? Oh, yes.
There's one on the right and a couple
Yeah.
on the left.
Yeah.
But when I find the dead ones, they're always white. Will these ones lose colour as well?
Mm-hmm. They're dead.
These ones are dead.
The white ones
The
without
white ones
the
are dead.
spines.
Yeah, but are they always pink when they're alive?
Different colors, actually.
Oh.
Some are white.
Yeah, but they don't have the spines, so I don't think they are alive. I don't think. Not sure. Wow, look at these crinoids.
Professor Tom, do you see a nice fishy in there?
You think he's listening?
Yeah,
Ah,
I think
he's
he is. Yeah,
listening,
he said
okay.
he's listening.
There's a lot of them on there.
Yeah? Nice.
Okay, so we'll continue.
Think yes,
Said he
yes,
is
please.
listening.
Yeah. Yeah, yeah. if you guys want to know, although we are actually here in the ROV room, you can listen to what we are saying, but also inside of the ship, people that are spread all over the labs, they can also listen to what we are saying. So we have Professor Tom, our fisherman, is in the main lab listening to us.
He's probably preparing his fish
Oh,
for dinner.
so
Wow,
many crinoids.
this is so nice.
Yeah.
Gonna say something in Portuguese, I'm so sorry, but Teresa, quando tu fores ouvir este vídeo, esta transmissão, por favor, faz um desenho desta rocha lindíssima, porque vale a pena. Eu não sei desenhar, já sabes como é que as minhas skills em desenho são bastante más. Por favor, desenha esta rocha que é lindíssima. I just say that, so I have a friend, she's an artist, she's Portuguese, and my skills in drawing are really bad, but I think she will love this rock, full of life, and I ask her to draw these. Or even our ROV pilots.
You see the whole Thurian?
Yes.
Oh, that's a
Oh,
very
the
cute
pinky
one.
Yeah.
Now just zoom in, please. It's nice. I don't want to sample in here. It's too shallow.
For scale,
Too deep, sorry.
For scale, how big is this cucumber?
So if you see the lasers, I don't see actually them. Are they on, Jessie?
Sorry, I turned off for beauty shots.
Okay, yeah. So the diameter, the 10? So it replies to your question. So it's 10 centimeters,
Oh, it's quite.
the laser.
What is the fish doing?
Yeah, look at that fish.
He's napping.
Professor Tom, anything that you want
Any
to say
comments, Professor Tom?
about
What
this
kind of
fish?
behavior is that?
I think he's dying.
Oh no.
He said it's maybe
Yeah.
he's typing.
Huh?
It's,
Should we continue
Oh.
with-
Blue screen.
it's an error.
Wow, so cool. Somebody in the chat says that Portuguese sounds so cool. Well, I don't think so. I prefer Italian. But yeah. I think Portuguese sounds like Russian.
Really?
Yeah. At least when Sveta talks in Russian, it seems to be Portuguese but backwards. Because I don't understand anything, but the sounds are the same.
Well, I don't know. I think I've been told Russian sounds a bit more like German.
German?
Just harsh.
No. Oh, harsh, yes.
Strong.
Okay, let's go on, please.
Bridge, you could resume that.
Resume the move.
You see the shot?
Is it that steep? Like, is it very steep right now? So, okay.
Yeah, we're going to do a little climb.
Yeah.
But it should flatten out pretty soon.
Yeah, we're going up, right?
Yeah.
Good.
You want yours even
Yeah? But shallower, because
There's
second dive is going to be
big
possible.
polychaete again.
So you need to.
But I
have
think we already
specific
sampled
numbers,
two.
more or less at the same time. That's fine.
Yeah, it's the same.
Cool. A sponge on top of a sea urchin. That's cool. Nice. Beautiful. Thank you.
Someone was asking us in the chat, how far off the sea floor are we flying?
Right now we went up so as close as we can. Yeah.
Yeah.
4 meters behind us, and we were half a meter off the deck. What is that?
What?
The long things, yeah.
Don't know. Can you zoom in, please? Here.
It's not a sea cucumber, right?
No, no, no. I don't know. Wow.
Polychaete. Oh, big ones.
Polychaetes. Interesting. Sea pens again. Oh, a sea mouse.
And that's a polychaete.
Yeah, I know. I just dissect one.
Most ominous thing.
Oh, Professor Tom, there's a fish.
Uphill, right? Yeah.
Yeah.
Yeah. Cool. Oh, interesting. Oh my God, look at those corals.
What is?
Can you zoom in, please? Really nice. Soft corals.
Are these the ones Aaron likes, or they're different?
Sorry?
Are these the ones Aaron likes?
Yeah?
I'm
But
sus-
we have collected already.
Yeah, I'm suspecting it's the same.
Yeah, think so. Thank you. Cool. I suspect there is another one there. Very nice.
I think on the first rock is also something. I think on the first rock is
Yeah,
also
yeah,
something.
yeah. Also, if you zoom in. Nice.
This is different.
Yeah, it
Going
is
around the
actually.
rocks,
No. It's just zooming. Yeah. We are not collecting. I don't think we have more space. Oh wow.
I mean, the coral quivers is space.
This is a coral. This is not. Cool.
Sorry, so the one on the left and the furthest right are corals?
On the right.
And what about the thing on the left?
I think someone was saying it's a sponge.
No.
No?
A sponge?
I mean, we collected one of these things earlier.
Here?
Yes,
These?
yeah.
Oh.
And someone was posting in the group that it might be a sponge, but
Oh.
not too sure.
Oh, there is a sea cube. Oh. It's lots of cool.
I've documented the rocks.
Yeah. Good. Thank you. Yeah, I also think it's a nitrogen zone. But another sea pen.
At which depth do you want to have your, like at which
As close as possible to where we're gonna have our push cords in the second dive, so towards the end. I think around 600. Before the transect, just here. Another CQ coming up.
I mean, you could say that we've seen them in the CC, and that's pretty 5,000 meters.
To answer to the question, how deep have soft corals like those have been found so far? Well, I actually don't know, but we found them in the CCZ at 5,000 meters. So at least to 5,000 meters they can appear. But I actually don't know if they can exist further than that.
I think it's a scuffle.
Mm-hmm.
What was
Octopus.
it? Ah,
Yeah, it's an octopus, yeah. Really going up. Oh, rocks. Sveta, rocks,
Oh, that's
rock,
nice.
rock,
Okay, good
wall.
rock. Quickly.
Yeah,
looks
cool.
laminated.
So it looks like some sort of terraces might have been cut out as this glacier was maybe retreating on the ice sheets. Or it could have been maybe like eroded in by this sheer like movement force of the ice.
We are at 710 meters, but we are going up, up the hill. Well, I am a marine biologist myself, and I do not have an aquarium. Have you?
My granddad had one, and I had to take care of it because he kind of rejected to do that. And then I was tired of that at some point, and I kind of.
I'm a diver, and I like to see the sea when I dive. I don't like to have the sea at home. Do you have one, Benjamin?
Octopus.
Yeah.
Octopus on the left.
Yes. Oh, and then nice sea star as well.
Benjamin, ¿tienes un aquarium a la maison?
¿Tienes un acuario en casa?
No.
¿Y tú, Marisa?
No.
No.
Me gustaría.
None of the marine biologists have an aquarium at home. But I bet Sveta, who is a geologist, she has a rock at home.
I do indeed have a
See?
rock collection of very rare minerals from Russia.
I knew it.
Yeah, my grandma was a researcher in Moscow State University in the geography department, and she used to do the old school remote sensing. So when you go on like aerial footage and take photos from above, of essentially kind of do like mapping from airplane, and she used to land to go to all these really cool remote places. So she collected this really beautiful rock collection of really unique minerals.
Nice.
This is a very nice looking thing.
And was that inspiration for
Cranoid.
you to go into the same field?
Cool.
No, but I did always look at these rocks that were in her kitchen when I was growing
Nice.
up, this nice rock collection. And then now she was kind of like downsizing, didn't really want the rocks anymore, so now I've got all these rocks. They're quite cool. I've recently moved house, so I'm just looking for the perfect display cabinet to put next to, instead of television, in front of the sofa for my rocks. Sometimes I bring them to class and I tell the student, talk to me about the rock for the next half an hour.
For your birthday, Sveta, I'll give you a rock
Thank you.
from Portugal. Yeah, you don't have one? do
Yeah, I
you?
don't have one from Portugal,
See?
this is true.
I
I
will
did
give
just
you.
collect a little collection in Chile just before my before I got on the ship.
I will
So-
remember that.
Send me one in post together with a birthday card.
No, I'll go to
You'll
the
come,
UK.
we can go to a nice music gig.
Of course. I always go to the UK for gigs. I have a gig session in August
Oh, excellent.
in Bristol.
Teresa is really into her music and gigs, and she loves Bristol.
Yeah.
Bristol's
Yes.
got a very good music scene in the UK.
Yeah. The Idols come from actually Bristol. It's a very cool band. So if you guys have never listened to Idols, please do. You won't regret it.
Teresa, do you play any musical instruments?
Yes, the bass.
Oh, cool.
I play the bass guitar, and I try to actually play the songs I like from my bands, but I can't yet because the bands I like.
There's a cup coral.
Yeah.
Should we take a close up?
And zoom in, please. Where is the cup coral? Oh, there. Yeah. Yeah. Also here. Yeah.
Oh, the
Yeah.
cute little mushroom-like
Yes.
thing?
Yeah. looks like an anemone, but it's a coral.
Mm-hmm.
Very confusing.
Is this for you, Maritza?
No,
Yeah.
Anna needs
Anna.
the only picture of the.
Cool.
Oh, okay.
Very nice. Thank you. Nice.
That's very cute. And this one too.
No, the idols, it's not.
So Teresa,
That.
you say, are you good at playing the bass, the idols, or is it quite
Wait,
difficult?
first you have to say that the idols are spelled IDLES, idols. Not the how is written. No, I cannot yet play idol songs because they are very fast, but I'm trying to. During COVID, I actually train a lot at home.
Oh yeah, I did that too. It's quite fun. Because sometimes late at night, if I'm still at work, you know, in the lab, I have an electric guitar, just put it on full volume in the department. And I'm really bad at music, so it's excellent, like Green Day, late nights, you know. I walk a lonely road late at night in that department. Next cruise, we take our guitars, make a
Yeah.
little band.
There's even a bass guitar here.
Is it?
Yeah.
Where?
In the
Oh my god,
what is it, backyard?
it's dangerous.
Bring it to the R&B control room for the next
It's actually
night shift.
really good for your brain because I memorize the songs and it's actually really nice. At the end of the day, you don't really read the song anymore because you know it by head.
There's another Capcom.
Yeah. Well, no, it's okay. It's fine.
So once we get to waypoint 10,
Yeah.
you want to look around for sea cucumbers?
I have to see how the sea bottom is. They don't really exist on these walls. So, oh, could you please zoom in these corals? Oh, wow. Big fish for Tom. Nice. Thank you. Wow.
It's a paracarid.
Amphipods.
It's an amphipod. Okay, but I think we have that one, no?
What is it?
Thank you.
Crustacean.
Mm-hmm. Yeah, almost there. Yeah. Oh. Still a bit.
Just about there.
Yeah. Yeah. Very nice. Oh, also some corals there. And there. Oh, another sea cucumber. The pinky one. Corals there as well. Here. Wow, this wall is amazing. Tom, fish.
Oh, there's
Oh wow,
what is that?
that's
Isopods.
cool.
Yeah.
Oh, can we
Where?
collect them?
Where?
The isopods on the red.
Oh, here.
Yeah,
Yeah.
because I think we tried to collect them earlier and they
Do you
always
think
jumped
you
off.
can try?
Yeah. Let's do it.
To
And since we're just got on our waypoint, so.
number 2.
Bridge. Collection, yeah, so
We
yeah,
go
come
for
to a pause.
2. You see?
Come again?
Can you come to a pause? You're going to collect through a collection.
I suppose.
Vehicle stopped.
Roger.
Mhm.
Oh, what are we collecting?
Isopods is a
Isopods.
kind of crustacean. They're sitting on the red
Yes,
thing
yes,
up
yes.
there.
Yep.
Oh, very
With the antenna.
What is it? Let me see. Oh yeah, it's there. Actually,
It's-
two, right? Three. Wow, a
4 or
lot.
5. And an amphipod.
Cool.
So whatever you can.
Yeah.
I think they're called Antarcturidae, but I'm not 100% sure.
Really nice.
Beautiful.
So yes, very nice. So let's go for it.
So we have.
Patricia has to buy me a beer. I don't like beer anyway. I like gin tonic.
Is this red thing, is that
I think-
coral that they're attached
Yeah,
to?
yeah, they are corals. I think so.
Remind me on two.
Yes, yeah, yes. Different species. Yeah.
That one is an amphipod.
Amphipod isopods.
So the hue on the rock we collected earlier is not magnesium, it's manganese, which is another sort of metal that's not too
Amphipods
far
and
off in the
isopods.
periodic table.
It's actually very nice.
You think it could be that one?
Hmm?
Alright. Are we all clear to swap our section?
Sorry?
Something. All clear to call our instructs thing?
Mm-hmm. Mm-hmm.
Antarcturidae. So it's very.
Sherman now.
Mhm. And what's the big like thing next to the red coral on the left?
Sponge.
Ah, cool.
And two fishes for Tom.
Professor Tom,
Three fishes.
update on the species, please. Maybe it looks similar to like the previous one, in which case I think Professor said it was Notolepis cortazzi.
Find a fun two.
I hope that was a good Latin pronunciation. Quite resistant.
Mhm. Oh,
Nice.
yeah.
Nice. Done.
So, section sample 2, Sveta?
Section sampler 2.
One
They're coming.
amphipod and hopefully more isopods. Get in there, get in there.
Nice.
Yes.
Thank you.
So don't come in.
Yeah, super cool.
It's
Thank you.
okay. Leave some. Thank you.
So three.
It's okay. Yeah, yeah, yeah.
About.
No, no, no. Leave it, leave it, leave it. We have enough. Thank you.
All zeroed on this section there.
Thank you so much.
So it's going in the section box 2.
Yes.
Yeah.
What's the species names of the fish?
The one previously, Professor reports it was called Notolepis cortzi.
Your Latin is great.
Thank you. I practice it in my Cambridge College at dinner when I have to read the grace, you know, sometimes.
Am I correct in saying that we have only 6 left in a section, or is that one also full?
I think it's all pretty much full. Yes.
I mean, here's nothing in 6, so I think
In
it should be
6?
Okay.
I have things in 6.
Really?
Yeah. Look. All full. Fish.
Anything else you guys
But it says fish question mark.
Yeah.
Okay.
No. Thank you. Yeah.
Oh, we have a correction from Professor. It is a nototeniniodis.
Millions of them.
Sure.
Yeah, let's going. Yeah. We are waypoint 10. Yeah. Or heading to 11. Yeah.
So, just for people in the chat, if maybe you've just joined us, so we have a Professor Tom on board. He's a professor of fish.
How often do you want to say
Well,
that?
because like there's a question in the chat they want to know. Just asked just now, so maybe they just joined. So Professor Tom, the fisherman, and he said these fish are nototeninoides. I hope that was helpful.
So we'll just put in a flag towards waypoint 12.
Yeah.
And then
Yeah, but so I'd like to see if I can settle between maybe 11 and 12. Let's see if it's flat. It looks like, right?
yeah.
Yeah.
Probably marijuana content
Yeah.
or maybe.
And we are getting shallower, so it's actually.
I can spell you the fish name, yes. It is N for November, O for Oscar, T-O, T-H-E for Echo, N for November, I-O-D for Delta, S. I hope that was helpful.
Yeah. Yeah.
Professor Tom, we have people very interested in your fish in the chat. There's a question, are those two the same species of fish? I think we might have gone past the two you're talking about, I'm afraid.
I guess you have to spell it again.
They for real? They want me to spell it again?
No, because you wrote it wrong in the chat.
Oh, they did. Okay, so let me just respell that thing. So it's N for November, O for Oscar.
Oh, you saw
Can you
that
go
one?
a
What's
bit
happening?
Cucumber?
Yeah.
Lower, and we are too fast.
So N for November, O for Oscar, T for today, O for Oscar, T for today, H for hen, E for echo, N for November, double I.
Yeah. With anemone and epibenthic.
Nice, thank you.
D for delta, A for alpha, E for echo. I hope we're clear about the fish. I'm afraid Tom is not in the room right now. He's just commenting, so we're translating what he's telling us about the fish. And this word that I've just been spelling is the family, not the species, which is like the thing before in biology. So species is the more precise identification.
The thing before is a good description.
Is it genus before family? Is it like species,
Family, genus, species, yeah, I think. But I'm also not very good in that.
But you will soon become the professor of biology and know the answer.
Oh.
So those fish, Professor Tom tells us, were they were the same genus but different
Nice.
species. So they were similar but slightly different.
Nice, another rock. Whoa,
Oh,
brachiopods.
should we collect? Oh, look at that.
No,
So
cannot
nice and
collect.
nice
Oh, rock
shell.
ahead.
Nice.
sea star flying.
Ooh la la.
Wow,
Shell
so
heaven around there.
so rich. It's amazing. So
Is there more fish questions?
No.
we are almost on top, right, of the wall.
Oh, sea star.
Fishy, fishy.
What is that white lily growing out of a rock?
It's a tunicate, a sediment.
Um,
Don't ask me what that is. I just know the name. Because Pedro said it earlier.
Benjamin, ¿qué es that?
Tunicate or sponge? I don't know.
Wow, too many corals.
Gracias.
Could you zoom in? Oh wow, that's so cool.
You want to zoom in?
Yeah, it's the same CVC, so if you can zoom, please.
Big sponge.
Oh wow, nice.
¿Qué es that?
Nice.
A tunicate. Probably.
Thank you. Wow. Really nice.
Ah.
Wow, such a nice sponge. And corals. Thank you.
I'm gonna have fun with that image material, annotating it in Beagle.
Mm-hmm.
Brachiopods, brachiopods, brachiopods.
And coral.
Coral.
I'd like some species identification of the brachiopods, please.
I have no idea. Sorry. That's all.
No idea, SP.
Yeah.
Maybe we ask Christian, he knows shells.
Yeah, he knows. At 5 o'clock you will ask him.
I will wake him up with an urgent.
Christian.
Benjamin is there. He's the replacement of Christian. Taxonomist. I'm not.
That Benjamin, you like shells?
Yeah, I like shells.
What kind of shells?
Gastropods
Oh.
or bivalves.
Brachiopods?
Brachiopods, yeah.
KS favorita shell.
Oh my god. I love your Spanish.
Ostiones, ¿cómo se dice ostiones?
Oysters.
Oysters.
Ostión.
Say it again. Oysters.
They're still going up, yeah? It's a rock. Nice. Really cool strange.
You're not gonna make it to your Holocaust point.
Yeah. Well, I think I will need to be here anyway.
I will also stay awake.
KS the snail?
Mm-hmm.
No?
Nice.
I think it was a small octopus.
Oh.
Octopus.
Mhm. Otherwise, I would wake up late. So it's not very good for the ships. We are in the same room. So even if I would want to go for a nap, it's not nice. I have to wake her up.
It's very considerate.
Hmm?
It's very considerate for me.
I'll stay up.
You see these ones? Did you see the
Yeah.
orange one? It's called spoonworm.
Worm.
Ecura.
So are you the worm professor?
These ones.
Amber, are you?
No, I'm not. I'm
No?
not. No.
What,
I'm not a specialist in anything. It's just I don't know. I annotated so many images, megafauna wise, that I kind of have an overview by now. But I'm really bad in like naming the parts of the animal.
That was like from now.
Yep.
So to pay the 11.
Yep.
So it's been a pleasure having Teresa here.
I need to.
Yeah, I mean, I can also just, if we don't see anything, we should submit it looks well flat. Yeah.
Yeah?
Just let me know which ones you want.
Yes.
We'll just
Yeah.
do 3 of those.
Yeah.
Fresh fruit.
No, it's only the follows through. Then we transect them.
Okay. Okay.
Yeah. Just go to the toilet.
Oh, me? Okay, okay. I think we've just got
Hey,
a
everyone.
change of everyone, apart from me, I'm afraid.
Good morning. Yeah, so we're just doing shift change for the science party. Sveta's going to stay with us, as she just mentioned, and we have Maritza and Anna coming in, and I'm Erin Easton from the University of Texas, Rio Grande Valley. And yeah, we're just going to continue along this dive. We started at around 950 meters, and we're working up to a little under 650 meters. We're supposed to have a few more hours of bottom time, but we're going to see how the dive goes. Sorry, there we go. Alright, so yeah, this is our 6th dive of the Climate Connections at the Icy Interface Cruise. And we are diving in front of the Bach ice shelf. We're on the eastern side of the Ronne Entrance. And we'll be doing one more dive later today for push cores near the top of this feature, so we can get push cores at the bottom and at the top. So we're just doing a little bit transecting here.
So an update for you, Erin. We in depth looked at this fish, and we have Professor Tom on WhatsApp commenting on the fish species. So we've had
Yeah.
a really long chat conversation with the audience about the fish as well.
All right, nice.
So
Thanks.
they're all experts now in the room.
All right. Good to hear.
No, all the people
Y'all
who
got
are
it.
watching us.
Then I don't have to say anything about any of the fish. You guys have it all under control. Perfect.
Because we don't.
It looks like we're doing a pilot change too. It looks like Claude and Jess are heading off.
You guys are going to. You have a trim on?
Yeah, we had Andy coming in to fly and Sunny coming in on the minutes. So we have Carlos down over on streaming.
We're just continuing transactions, so. See you guys. Thank you. See ya. Thank you.
So I think we still have a few collections left to do before we leave bottom and finish today. We're going to be collecting some more holothurians for Teresa, and then
Just
keep our
some over
eyes
there on
peeled
the
for
left.
some rocks and corals. What's on the left?
Some holothurians. We didn't see too many actually in the previous shift, and now that Teresa is left, we seem to be catching.
Yeah, I'm sure there's a few of them. Yeah, we can keep going. We'll keep transect mode for now. She said she'd be back and let me know. So she should be back shortly. She'll message us if she sees something super exciting.
Where's your RV?
So yeah, Anna, do you want to say anything about what you're doing on the cruise? Can I update the
Using
cruise?
this one.
Yeah, use that one? Okay.
Okay.
Yeah, we can hear you really well.
Alright, perfect.
Hello everyone, I'm Anna Maria Damo. I'm working at the Nord University in Norway, and here in this cruise I'm working with eDNA, the environmental DNA, and the microplastic. What we want to do is try to first characterize their habitats. And since it's impossible to collect all the organisms that we see and during the time of the ROV. So the idea is to collect the water and extract the DNA from the water and try to identify all the organisms that are in this habitat. And in meantime, we analyze also all the ROV videos and images to actually match with the result from eDNA. And then, because characterization of the habitat means also impact, a potential impact from human activity, we want to also notice if there is any microplastic in the water at the bottom that could actually impact the animals that live in the seafloor. So this is exactly what we are trying to do in this ecosystem that is in Antarctica, that is quite difficult to reach and to actually cover and survey with a sampling in a big area. Oh, nice! Sorry. That was a nice octopus there, sleeping, almost like us, but
Like us in this room at this time with the night shift.
yeah. This night
How
shift
many
is,
hours a day do the octopus sleep?
maybe it's like a cat, can sleep all the time, except when he needs to eat.
It's got nice color.
Oh, yeah. Look at the eyes. Oh, so beautiful.
Oh, it's moving. It's moving its tentacles.
We're waking
Because
it up.
of the light. Uh-oh.
Wow.
Nice.
Uh-oh. That is a nice...
And is it Friday now? So is it Octopus Friday? I know you guys have probably seen some, but
it's Friday. You're right. It's Friday, 24th January.
it's Friday for us at least.
Yeah.
Wow.
Nice. That's hopefully a nice highlight video for Alex. So far all the octopus we've been seeing have been sleeping. On the side, at least that I've
He's
seen.
waking up. Look, wait.
That's cool.
And you see? It's changing a little bit color there.
Yeah, it's getting a little more like fluorescent.
It's changing color.
Yeah. Oh, yeah.
Wow.
Nice. Please flag that for Alex.
Oh.
There it goes. Nice.
It changed color. Okay. We actually.
Yeah. Alright, we can go back to trans.
Look, Sveta, good shot. She's shocked because she's
shocked and also happy captured the video of octopus.
yeah, in the Octopus
2 hours
Friday.
already, we haven't really seen any, or we've seen some small ones.
Yes, I'm sorry. I wish I could tell you which species it is, but
Oh, no.
I think the ones we've been seeing have been. Someone put it in a chat at some point, but I don't know if it's the same one. So. Yeah, so let's see. What's your favorite thing you've seen so far, Sveta?
Oh, Sveta.
Oh, I saw some beautiful rocks.
A part of that?
Well, no, this is true. So we were discussing a little bit about rocks with those who were online earlier. I'm not sure how many people have recently joined, but I'm a geologist in the wider sense of the word, and I like rocks. And I have a beautiful rock collection. Lots of
That
this.
is
That's a nice rock.
Yeah.
Let's document the rock
Nice.
and not the animals.
I think there's an octopus. You can go ahead and look over here. I know since we're ahead, but see, we see another octopus and a rock, and a crinoid, and an urchin.
So maybe it can be documented as a bio.
Yeah.
Well, that's Maritza's job. I just document the rocks. But we did see these beautiful rock terraces, and they were really full of really cool, like loads of different things, including some nice brachiopods. And then we had this thing, I took a photo, but I forgot the name of it, so maybe you can remind me for the audience. examined biology.
The pink one.
And we were taking dinner for Patricia. These
You take
prawns.
isopod on the top of octocoral.
Oh, it was and then this beautiful.
No, this is octocoral. This is the isopod, and this is, I think, an ascidios. Looks like.
So I'm just showing the picture to Anna, who's a professor of biology.
No, professor, but
Oh yes, we are all professors in this room. Don't be shy. We had Professor of Sea Cucumber, we have Professor Fisherman. Now you're a professor of what's your favorite organism?
Coral.
Oh, but no, that's
But
Erin.
Erin, that's why-
Erin is the coral professor.
So we do another general cnidaria.
Of what?
Cnidaria, that means
Cnidaria.
Cnidaria, the cap corals.
The cap corals, professor of cap corals. So I think it's very nice.
The cap professor.
So I was just showing Professor here next to me a picture of the beautiful pink coral brain-like thing that we saw earlier when we were sampling those little prawns.
Prawns. Oh, you know. So yes, that is the favorite thing.
I'm
So
sorry,
someone's
Professor
asking about
Erin.
this rock. No, you're good. Someone's asking about the rock.
This
So
rock?
Probably a drop stone.
What do they want to
think?
know about the rock?
Like, it looks like it's kind of split or whatever, I think. But I don't know. Where do you think this came?
Yeah, I think it's probably, drop stones. So drop stones are these things that basically get spat out by the glaciers at retreat. So as it melts and sort of retreats back, it breaks off the ice shelf and moves back further into the continent, and then like just spits out these rocks. And the reason it even has rocks is when the ice moves along the continent, essentially it's a bit like a river, it actually floats even though it's solid. But the sheer strength of this moving ice, because if you imagine the strength that it takes to like ram through the rocks that are underneath the ice, it picks up all sorts of rocks and sediment and whatever is underneath the kind of like riverbed of the glacier, and then once they start melting and the glacier retreats, it drops these things into the sea floor. And right now we're in this Ronne Entrance, which probably deglaciated.
We'll continue transecting now.
It was definitely glaciated last glacial maximum, so last ice age around 20,000 years ago. We don't know exactly when it deglaciated or when the ice retreated, which is partially why we're here from Geology team. We want to kind of try and understand the time frame of retreat in this area, but so that this rock had been dumped in the last sort of, I would say last 15,000 years or so on the sea floor by the retreating ice sheets.
Oh, cool. That's why we see sometimes in these soft sediments a lot of rocks.
Yeah, just like spit, yeah.
Spit rocks. So that
And for
was,
the octopus, it is a different octopus. So I'm just answering a question. You guys can continue your conversation. Lovely.
No, that I was saying that's at the end was an octopus.
Okay.
Oh, there is something on the. Is a Dumbo?
So for people in the chat that are interested in the cocktail sauce, I would agree. So I've documented the prawns we collected as a dinner for our chief scientist. Because our chief scientist, Patricia, she's a prawn expert, and these prawns actually have, sorry, can you give us the correct name that they're called? And what is the other one? There's two that I document. There's the other isopod.
Isopod and amphipod.
Isopod.
And yeah, so
amphipod.
those things. So we collected all those things, which I thought would make a really nice dinner.
I'm not so sure. I mean, they are bottom feeders.
You don't think they're very meaty?
I think they might taste a little bit like the dirt. But I'm not a seafood fan, so. I'm making his paella. There you go.
Is it an underwater and glacial erratic stone? Honestly, I don't know. I'm not actually a rock expert. I work with dead shells. But yeah, it's definitely of glacial origin, because this is again would have been spat out by those glaciers. So yes, it could have well been like picked up on land and broken off, and then deposited in this area.
Awesome. And I guess to recap earlier in the dive, like we first started this dive and we were on shift. Most of us were on shift at that time too. And we started out seeing a lot of sea pens. And so Maritza works with sea pens. So Maritza, do you want to tell them you can tell them in Spanish about the sea pens that we were seeing earlier?
Hola a todos. Bueno, en este buceo creo que me asombró mucho la cantidad de plumas que encontramos. Fue un campo de umbellulas, que son este tipo de plumas de mar que parecen un paraguas o algo similar. Y la verdad fue muy bonito porque yo nunca antes había visto este tipo de umbelula. Era diferente a las que nosotros vimos en los cruceros en los montes submarinos, así que fue muy genial.
Yeah, so Maritza was saying that at the beginning of the dive we saw a large field of these sea pen, the umbellulas, and they kind of look like an umbrella, or like a parasol, so they're really beautiful, and we haven't seen that quantity of sea pens before on this expedition. We're on dive 6. And then also she was commenting that yeah, this is like Maritza's 5th expedition and like 13 or 14 months on this ship, and she hasn't seen anything the same species on any of the other expeditions, so it's a distinct species. And yeah, we collected a sample earlier on the dive. And so we're also seeing like a different, oh yeah, this is the anemone we were seeing, the tube anemone we were seeing earlier in the dive. They were passing these kind of white anemones. I think we collected one of these sea urchins the other day on another dive. Lots of sleeping fish. Let's see. Got just a waypoint and a half to go. I haven't heard from Teresa yet about which sea cucumber she wants. But she said she'd be back. Looks like we have maybe another octopus or a rock coming up. I don't know. Yeah, I think it's the Dumbo octopus. So this one, or is it the same one as before? No, this one's a Dumbo. Yeah, this one's a Dumbo octopus. So this one is different than the species we saw before, and this is either probably a Grimpoteuthis or an something like that, Optoteuthis. And probably, yeah, we gotta sit down and get a shot of the Dumbo octopus. So yeah, we've been seeing quite a few octopus on our dives compared to the other cruises I've gone on. Sometimes we occasionally would see a lot, but we're pretty consistently seeing octopus on each of our dives, so it's kind of nice.
So the ice would not necessarily pick up fossils and the commons that we have from millions of years ago, but what happens at the seafloor generally when these sediments accumulate is kind of like a window into the past. You get new accumulation layers on the top, and the deeper in the sediment you go, the further back in the past you go. Sometimes millions of years, sometimes only thousands, and sometimes only decades, so that sort of depends on, and this varies depending where you are. Built up would have built up since the last ice age, so around that 20,000 sort of year mark, whenever it began to go season.
Okay, actually, can we stop?
So if we collected a long core of sediment, if you imagine a long tube filled with sample, that would all be probably fairly recent with the Holocene, so the warm period since the ice age, the last 10,000 years.
1B. there's 3 right here actually, all kind of close together. So one's a little bit bigger, but scoop them up. Yeah. They're just repositioning to not sit on the Dumbo octopus, so.
Good. Yeah, you can come to us up here, we're gonna do some sound.
1, 2, 3, 1.
Teresa's asking to put it in the empty bio box.
Yeah.
Yep, there's 2 empty bio boxes. Yep. Whoop, there rolls away one. Yeah, with the scoop, and then putting it into one of the 2 empty bio boxes, 1B or 2A.
Do the octopus have any natural predators at this depth?
Good question. Not sure. I mean, yeah, I would think they have something, but I don't know what.
Lowers off.
Maybe big predator could be the
Yeah, some top predator. But, I mean, you know, this part where we're at is covered a large part of the year in ice. And it's really just a few months of the year. So how that changes the community, I'm not really sure.
unless they are the big predator.
I mean, where we're planning to go in our next station, so in a couple days, once we map it, we'll be diving at an area that up until two going on 3 days ago was covered by sea ice. So, yeah, I think a lot is changing in this part of the world right now with all the sea ice breaking apart, melting, and these communities are changing.
Are you rich?
So.
Do you have one of the MTs down there?
Oh.
The only one.
The only one.
You just ask them to give me a call on the phone.
Here, you want?
Heading that way.
So I think the octopus was sleeping and his eyes were mostly closed. In here, we're not quite deep enough where most things are lacking eyes. So we are now at 600 and some meters depth, and actually 663 meters. And at this depth, there are, in clear water in particular, you can get extremely low levels of light. The large eyes can actually pick up some of that light and the bioluminescence. So it's usually a bit deeper than this, then we start seeing a shift over to organisms that are missing their eyes. So, yeah, but it is quite dark down here. Not enough light for us to see at this depth. And sometimes our lights are so bright, for some animals we're basically blinding them, at least temporarily, because they're adapted to seeing low lights and these little flashes. And so shining this much light will temporarily blind them, just as if you look up into a bright light you'll be seeing spots and everything else. Same thing essentially would happen, or worse, for the animals down here. This is where I need Fernando to answer all these octopus questions for me.
Fafa.
Yeah, I need Fafa. Where's Fafa?
I don't know.
Fafa's sleeping right now.
Yeah, that.
It's a talking to
Is a Spanish researcher working on octopus too.
Oh, you're going for the far one. Sorry.
Yes,
Sorry.
that's for sure. This area is quite productive, although this site compared to the other side of the water mass that we're in right now, or the
You guys know anything about the quarry? Where's the waypoint location? For the quarry
for
in the
dives.
morning?
Gravity core?
Yeah.
Maybe we can pull up the dive plan for the next dive, and then we should have a waypoint.
Yeah,
in there.
very
This, are we talking about gravity core?
Gravity core.
Oh, gravity core. That I
Yeah.
don't know.
Because we don't have a waypoint. We don't have anything.
Okay.
Are we doing gravity coring at the station?
I don't know. It's on the schedule.
The daily plan. So
Gravity core number 5
It's
and 6.
3 at 7. So we don't have locations for any of those.
And
they
this?
haven't shared that with us.
Yeah, I think there's some, I'll just compile and check.
Okay.
Yeah,
Where are we putting this guy?
Either Biobox 1B or 2A, which everyone. Oh wait, there's a fish in 2B, so we better do 1B. UTC. There's UTC. Yeah, we're just collecting the 1B. 1B is empty, so. Yeah, so we're just collecting here 3 individuals of the Scotoplanes sea cucumber, so that Teresa can do a comparison from deeper communities to shallower communities at each of our study sites. And yeah, I don't know what kind of, honestly, I really don't know what kind of protection, if any, the octopus have to lights. I would have to ask someone who works with them. I don't know enough detail about their eye function, but they do tend to not react too much, in my experience, to light when the ROV is around, especially when they're sleeping. So.
BB 1B.
Because upstairs it looks like the same.
No.
Yeah, yeah.
Only one people.
I can wait. No, tengo miedo.
Look at this.
No.
Yes, educate us from home. What you find online. And yeah, it's really, I was starting to say, it's really different, the productivity. So we're at the Ronne Entrance right now, and we're on the eastern side. Earlier we were on the western side, and at our last station, and there is a substantial difference in the amount of productivity in the water column. So there's less productivity at this side than there was the other day at the other station when we were on the western side. And so we have some of that information from CTD data and then just some visual observations where when we were putting the ROV in the water was quite green near the surface. And here we didn't see quite that greenish tinge. We also have noticed difference where the top layer on the sediment from that productivity raining down was more green at the other station than this one. It's like a gelatinous organism jiggling on a gelatin.
Como si lo inflara. Como si lo inflara.
Oh my gosh.
Uy, uy, uy.
No, so we don't, these boxes are not set up for pressurization, so any of the animals that we collect at depth are brought up and the pressure change, they experience that pressure change. Which for a lot of organisms that are like invertebrates, for example, that pressure change isn't a big deal for them, but for others it can be an issue. There he goes.
Okay.
You can do it.
Yes.
Come on, backflip. Got it.
Great. Great.
Nice.
Amazing.
Oh.
Okay. A little bit.
Yes, the common name is a sea pig.
Snow.
Waiting for the sediment to clear a little bit so that you can see this third one. Oh, there we go. The sediment's clearing.
Ready?
Yes.
We're gonna talk about the taxonomy next. Someone in the chat asking about if you find a new species, how do you give the name?
Oh, okay. Thanks. I didn't miss
You
that.
use, I don't know, your name or someone that you know.
Yeah, when you find a new species, there's a lot of work on describing it, and once that's done, you get to name it. But there's rules. Like, you can't name it after yourself if you're describing it. You can name it after someone else. friend, family even, but you can't name it after yourself. You can name it after like a feature it has, or there's been some named after famous people. Yeah, some named after characters and films or books. So yeah, you just have to follow the naming conventions, but you can put, within limits, the name that you choose.
Do you think, I have a question. Do you think it would be better to use feature or people to give a name? Just to,
If you were here to give a name? Sorry, I can't hear you.
I'm sorry.
Do you think that would be nicer to use features so it's much easier to recognize, to link the name to the species, or name
Thank you very much.
to the people, like, I don't know.
Yeah, I mean, I think the features are kind of nice to name them because it makes sense. Some people, a lot of them are named after regions, which is nice to a certain extent, like the region where they're found or collected, but it also gets a little bit confusing because you end up with a whole bunch. For example, I've done a lot of work in the Salas y Gomez Ridge, and there's a lot of them called like Salas y Gomencia, or like so. And so you have a lot of similar names. Alright, great. We can go ahead and continue on the transects. Hold on, I can't hear. Okay, that's fine. Okay. Alright, onwards we go.
You like it.
Hello everyone, I'm back.
Hey, so did you find out the gravity core waypoint?
To find out the gravity core location.
Alright.
Way. It was a bit awkward because we had to go wake the co-chief scientist up, which I haven't done before, and then
Where's your RV?
just 1 minute.
Okay, you could start the bus and move to the next RV.
So yes, I had to go wake up the co-chief scientist to confirm something, and that right now we're at 2:30 in the morning. So it was a bit awkward just because it's like 2 men in the cabin and me just kind of trying to go in there. So I took Professor Tom for moral support and asked him to go in first because I was too scared to go into this man cabin. But we found out the gravity core location.
That is the most important mission.
Exactly. I suppose because it's difficult. We had 24-hour operations on the ship, and sometimes, like today, we have very busy schedule all through the night and the early morning, and of course people still try and get some sleep between different operations. So sometimes you have to go and try and wake them up and find out information in the middle of the night, which I'm sure is not very appreciated on their side.
Yeah, I guess lesson learned. Before I go to sleep.
I'm sure they do, yeah. Tell the bridge the coordinates of the gravity core.
Don't forget, so you don't get interrupted sleep. Yeah. There are things.
What have I missed?
Jiggling sea cucumbers on piles of jiggling sediment. Yeah.
Did you
We collected all 3 for
lots of
Teresa.
samples?
Yeah.
I hope Teresa is happy. Professor of sea cucumber. Very experienced sea cucumber pathologist. For those that have missed earlier, we decided that after the surgical procedures, the sea cucumbers don't have a very good survival rate. So I'm afraid Teresa has been downgraded from surgeon to pathologist. Or maybe upgraded, depending which way you look at it.
Let's see, the current water temperature, actually, I think our sensors aren't connected right now, but I believe it's probably around 1.5 degrees Celsius. It's about the temperature we've been at. Might be a little bit off from that, but yeah, I know. Yeah. I mean, it's kind of obvious. Yeah, it's just our CTD, which is the connectivity temperature and depth sensor on the ROV. There's just a connection issue with it on this dive, so yeah, they just had to turn it off for this dive, so we don't have that data coming in, even though we usually do. We do have other data from other sensors, but unfortunately we don't have our temperature sensor working right now. So we're nearing waypoint 12. What now? 1.24. Alright, they just pulled up on one of the other sensors and looked at it. The temperature is 1.24 degrees Celsius. So.
The new expert. Thank you, that was.
It's actually, well, I don't know, is it still snowing? It was snowing earlier when I was working out. Yeah. Yeah, right now it was kind of nice for a little bit. It was sunny and snowing at the same time. It was really pretty. I was watching the view from the holodeck area while I was going for a little run and bike ride between my
Oh, we
shifts.
saw one of these worm things earlier.
But they make me. This is third time.
What is it?
Ah, no, I don't know. It's just a worm. I say that. I'm not that person. Oh, worm is just a worm. A long one, actually.
Yes, we saw one earlier. It was unfolded and it was all like flopping. It was quite extensive.
Yeah. And we haven't even collected that one. Maybe our colleagues don't know that one.
I would say it's the same worm as before, but I really don't know my biology, so don't quote my word on that. That one was also long and wide. This
Yeah,
one
it's
is
the
long and
same.
wide, I think.
We saw it from
You
the...
saw? Oh, you saw
yeah,
it from the
so
lab? Oh, cool.
It was the same. But now name idea.
There's some more sea cucumbers.
Yeah, still seeing a couple different. There's that other type of sea cucumber we were seeing before. Yeah, so actually there's a lot of. Yeah, let's go zoom in on that. Give a nice.
Cool.
Get some lasers on it, and then we can get a glamour shot of it. Yeah, so there's actually a lot of redundancy in terms of sensors on the ROV and other equipment. It's just, in this case, it's not all set up for us to easily view in the room, but we're able to pull it up later or switch screens. And so when one sensor's down, like the CTD, we can usually get all that information from other sensors on board. Alright, yep, we're good. We gotta continue transecting. Thank you for grabbing. Nice image.
Did you just get woken up? We collect to do some prawn cocktail. Sorry. Erin, what corals? I heard you collected some really cool corals earlier in the dive.
Yeah, so I apparently the corals like to show themselves while I'm sleeping. So, but from the images that were taken, it looks like we got a couple primnoids. I think we got our Gersemnia again, which is nice. So we have a comparison from two different stations. I think we maybe got an acanthogorgid as well. But I would say I think they have 6 collections, and I would say at least 3 of them are species we haven't collected before, based on the images I saw. Maybe 4. So yeah, we saw, So it's been really interesting. Each of the dives has had different species largely, so it's been really, really interesting so far on this expedition.
Where's your RV?
But there's a lot of sediment, so the corals are often few and far between. So whenever we come across some rock outcrops, we'll tend to see some corals.
Where's your RV? Okay, and you waypoint. No worries.
Did you tell them about your 3-meter gravity core?
Oh, I haven't actually yet.
Yeah.
So we've been collecting these gravity cores. That's essentially a bit like the push cores that we collect off the ROV on the dive, but we just overboard kind of throw this really heavy thing into the seawater. What is that?
A sea-pen.
Oh.
Really nice big one.
Oh, that's a nice one. How tall is that? A meter?
Probably.
Wow. But yes, so we've been collecting these cores where we just chuck this heavy metal thing get over from the A-frame, basically, from the behind at the back of the ship. The core liner is up to 3 meters, so we can in theory collect 3 meters of sediment. First 2 we collected were at 1.6 and 1.7 meters approximately. And yesterday we got really lucky and we actually collected the full tube core liner. It's 3 meters long. And in fact it was so long that we had to cut it in half to fit it in the fridge. And then Anna here had a lot of fun. We took out this special book we have that's called Munsell Soil Colour Chart. And this is a book that actually is quite expensive. Surprisingly, it costs around $300 because they have these hues of basically colours of soil, so all the shades of brown, grey. And what you do is it's a bit like choosing wallpaper for your bedroom. You sort of walk along your core, along the whole 3 meters, and document the colour changes that you see in the core by putting this palette against the core. Now, as you can imagine, that's quite difficult because there's a lot of shades of brown in this book and only one core. So we had quite a bit of fun yesterday just trying to document the colour changes in the core.
Yeah, it was quite interesting. It had like grainy pieces in there too. So like you could see it was kind of yellowish near the bottom, if I recall, and then it had this quite long area, maybe 10 or 15, 20 centimeters of like where there was like pieces, like you just see like chunks of little pieces of rock, like little gravelly or slightly smaller pieces.
Yeah,
it's
and
kind
then
of
the
brownish
middle
red
section,
near the top.
Yeah, and then the middle section was kind of like bluey green, and you start having a bit of paranoia whether you've got color blind because you look at this book with all the colors that look the same. But Anna, I think you really liked it, right? The
Yeah.
little color book.
It was super.
Super fun.
Yeah.
You learned some geology.
Indeed, for
Yeah, how
1
was
minute,
that?
I became a geologist. Just 1 minute. I code the color. Every color has this code, and then you have to go through with this book through the core and see the color. It was really fun. But for 1 minute, I became geologist. Then I.
No more.
Guess your eyes have to get trained to seeing all those differences in colors. I know when I first went out there, I was like, well, it kind of looks uniform. And they're like, no, it's like way different. I was like, okay, let me look at it again. I'm like, oh, now I see.
Next time, bring your binoculars.
Just like you have to like train your eyes to see the differences in the corals. I'm like, no, that one's different. You're like, what?
Yeah, exactly.
Octopus there.
Oh, that's a cute little octopus.
Mm-hmm.
And small.
It is not blending in very well, though. Wow, that's cool, though.
Why is it so fluorescent?
I mean, the other one went fluorescent, I think, when we started to aggravate it. Yeah. Maybe it's on the watch out, but I don't know.
Maybe it got scared. Oh yeah, it's starting moving. Probably doesn't like the light.
Alright, we're good. Thank you. We got sleeping octopus, sleeping fish. Here we are.
Everything is sleeping.
And we're still awake. Dreaming about our sleeping.
I'll probably feel that way in a couple hours. Right now I feel pretty good. I went
Wait
to
till the samples come on board and then it'll be really fun and the cold really wakes you up.
Yeah, it's true.
Really cool.
We are
Really
lucky.
cool.
The ROV is brought into a hangar, so it's all kind of closed off, so it's cold, but it's not so bad. Especially on the day when we were having like 30-some knot winds when they were recovering the ROV and snowing. The poor ROV pilots and the ABs were, bosuns were all having to deal with the wind and the snow, but we get to stay all safe and inside.
It does still get like pretty cold in the hangar though after a while if you just stood there like
Mhm.
cutting up some sediment for a few hours.
Yeah.
Which is what I do every time the ROV comes up. Basically, I'm a really annoying person, so I like my sediment cut up into 1 centimeter, this all 4 cores. So it takes a little while, and every time I'm asking, anyone want to learn about sediment? And slowly the amount of volunteers is decreasing, or very quickly I should say, to help me cut up the sediment and bag it all up into individually labeled bags. So it does, after a while you feel the cold. But yes, I agree, we are lucky to have the hangar. But it's quite interesting actually, because in Antarctica, sort of beyond the polar circle, we essentially have no darkness here. So that means every time you do go outside in the middle of the night, not only do you wake up from the cold, but you also wake up from the sunshine. Sometimes it's really sunny at like 2 o'clock in the morning.
Curious.
What is this guy?
It's a mollusk. And on the top, it's an enemy.
Yeah, it's a mollusk
And
wearing an
shell.
anemone.
It's really nice color.
We already collect this one.
I don't know, like, is this a mollusk that's on the, like, do you think we should get this? They want mollusks.
Erin is asking
Oh, sorry, I wasn't.
if this needs
The
sampling.
thing is, like,
No, I think they already collect.
they already collect? Okay.
Yeah, Kristen
Okay,
already
thanks.
collected this,
Alright, there's no wish
at the
list.
beginning.
Great.
Let's go ahead and continue. If they want it, then we'll keep our eyes peeled for it another time. Most of their containers are full. So, drop stone. I think it's more I'm getting hungry than anything else. I'm not tired yet, but I'm getting hungry.
That's why I had 2 big steaks at around
We have
midnight
chocolate.
just before.
Oh, you want this one?
Maritza's prepared for the night shift.
We also have other chocolate here.
I'll take this one. I'll be good with
Other
this.
choices.
Thank you.
We got
Wow.
our chocolate. This time with spicy chocolate.
Spicy
What
chocolate.
am I opening?
Maybe a spicy octopus salad with a prawn cocktail sauce.
Okay, here we are preparing the dinner. From cocktail water. You wanna?
Thank you. Oh, it is actually spicy chocolate. Well, what is
Yeah.
that black guy?
It's
This
an
one?
urchin.
thing.
Sea urchin.
Yeah.
What is that black guy?
Yeah, it's like my soul. I like black rocks and black t-shirts and black hair and chocolate.
No.
No, we're not seeing the comet. We actually don't get. We have daylight for 24 hours a day right now, so it gets like a little less bright out, but the sun doesn't set. So we definitely are not seeing. Like, we can see the moon. Like 2 or so in the morning, the sun's still up, and then the moon is up by then. So we're after like sunset time essentially, but the sun never sets. Yeah, we're at, what are we at? 72 or 73 degrees? 70?
Oh, look how still it looks outside. We just got camera front view of the front of the ship, and also this ray is very nice.
It is. Yeah, that's what I'm saying. Like, look, it's like snowing and it's like half cloudy, and it's beautiful out. You know, it's what was on the TV when I was doing my run. There were too many people in the gym. I couldn't change the TV control because I'd have to like, well, shockingly, at midnight there were 3 of us in the gym.
Outrageous.
I know, right?
I think it's because tonight we actually have more people on night shift than we normally do. So maybe people are just trying to get some energy to kind of stay awake for all this late night sampling.
That was exactly my plan. I was like, well, I have an hour and a half break.
I went at 10 a.m.
Professor of sea cucumber, Teresa, is back in the room with us. The cucumberist, someone called you from the audience. I quite like it, cucumberist.
Went to the gym at 10 a.m.
She went to the gym at 10 a.m. today.
See, that's when I would normally go, but now that they changed our shift, it doesn't quite work out for me anymore. I woke up at 10 a.m. today and I was like, oh, I have to work until 4 a.m. At
So
least.
to answer about the gravity core, no, but we will be deploying a gravity core straight after this dive. So right now we're around 3 o'clock in the morning, 3 300 hours, and we plan to deploy the gravity core around 6 o'clock in the morning, so 0600 hours in about 3 hours time. And I think we forgot to specify the exact coordinates of where we want that heavy metal tube to be dumped into the sediment, essentially, to sample us some the gravity core. So I had to go do some investigating from the sleeping geologists of the recommendations for the gravity core. So we have the captain basically just wanted to know the exact coordinate location, and to determine that we basically just use this something called sub-bottom profiler seismic data. So essentially this thing, it kind of uses seismic waves to shine through the first few layers of the sediment. Oh, it's snowing. But yeah, so the seismic sub-bottom profiler shows you the seismic data, and basically you can see these lines of like the thickness of the sediment, because a lot of the areas around sort of glacial, exoglacial kind of terrains, so you basically just do not have much sedimentation here. So you kind of just need to look for these little pockets where you can collect your gravity core, because some areas is exposed bedrock. For example, like when we were diving or flying over earlier, you could see there's just rock sticking out with loads of interesting biological specimens. So we do not want the gravity core to hit that, because essentially what happens then, it comes back as this banana. So it's bent and then it's broken. Professor Tom?
Yeah, is this one of his like wish list fish here?
Professor Tom, quick,
Yeah.
please assess the fish.
I think
It looks like a snail, so I think it is a snail fish.
I think so.
If it's snaily in shape, then it must be a snail fish.
It's an
No,
eel
the
pout.
snailfish has a
Okay, it's an eel pout.
It's a eel pout, but they already
Oh no,
collected
this is an eel pout. And
because they're not snailfish.
let me know if you would like me to spell that, but hopefully not this time. In terms of samples for gravity core, so that just depends on how much time you want to dedicate to this particular piece of equipment, because we have a very wide program on this cruise, because we have a very multidisciplinary team. So gravity core essentially is only of interest to a few people. So we don't tend to collect more than one per station. Yeah, but sometimes you have a geological crews which just deploy gravity core kind of like 1,000 times, you know, every location they find just deploy the gravity core. So it depends on the objectives. But I think the biologists on board would be very upset if we just deployed gravity core and not the RAV.
And I think everyone at home would be a little sad too, because they don't have like a live stream on the gravity core.
They could have a live stream of the gravity core, but it's quite boring. Most of the time you just see the wire of the winch just go at spit. It's just, you know, lowering it or picking up from the depths.
Mm-hmm. Yeah, and I know I'm not able to row on the outdoor lounge right now outside. The outdoor rower is still there. There's a question in chat. I'm well known for trying to row.
I'm really enjoying
feel like
this
I'm rowing myself.
gravity coring situation in the chat as well is
Yeah.
excellent. Thank you. So when collecting a core, as the water drains out once it is at the surface. So the whole idea is that this essentially is a tube that just goes into the sediment, so it comes back without water but full of sediment. What we see with push cores of the RAV, we're actually kind of collecting two different types. Some we ask our pilots to leave some water in them, because the biologists are interested in that water for some reason. Don't ask me why, because I think sediment is the best part. So for my cores, the pilots are very nice and they just stick it right into the sediment. So I have minimal water and a lot of sediment for my geological analysis.
That is also the difference between your push core and the other push core.
Yes, exactly. And my push cores are actually a little bit longer than all the other ones that everyone else gets, because again, we want that long sediment. I'm afraid I don't know much about engineering of these corers things, but essentially if you have like a 3 meter core, as long as there's 3 meter sediment, the tube and the weight is like heavy enough on top to push it in. So it kind of just works with gravity, as the name gravity core. It's just got this like loads of weight on top. So the one we have on board right now is 240 kilograms, and you chuck it off the winch from high above. And just with the force of gravity and that weight, it basically just rams into the sediment and just collects up to that 3 meter. We could make it a 6 meter gravity core. We've actually got that possibility on board, but we're a little bit worried that we'll just then break it and then we won't have gravity core anymore, so we have not done that. You can do a lot of different things with sediment. For me, like some people actually study the sediment, so like size of sand grains or color of the sediment, as we were discussing earlier. There's whole scientific papers about the color of the sediment changes. Sometimes we do a CT scan as well, which is quite funny, that I took some sediment cores to the local hospital in Cambridge. And someone in the hospital in the CT, like some patients, suggested there was some pythons and coffins. So, you know, people get a bit weirded out sometimes by sediment cores in the CT facility. But essentially, I look for these little microscopic shells, and that's analyze their chemistry. Professor of sea cucumbers, please can we have an update on this situation? Would you like to tell us what it is? Sea cucumber? It's a sea cucumber.
It's okay. I'm talking with. It's okay. I got it.
That's a nice sea cucumber. Together with the fishy fish.
Aww. Cool, thank you.
Do you want this one? Looks so nice, like the little spikes. It kind of reminds me of some like dinosaur ancient creatures. You don't think so? No. That's the answer from Professor Teresa.
Yeah, we can keep transect ahead a little bit. We can always, if they want us to change directions, we can turn off, but I think this ridge line, keep following it. We're seeing some different things here anyway, so, which is kind of nice. Nice, healthy looking sea star.
Yes, I agree. It does look like a punk rock holosaurian. I think that would be a good name for an album. Our future for our music band. Is anyone on our cruise interested in the sea urchins?
Yeah, so I think just Christiana's collecting representatives for museums, but no sea urchin expert on board on this cruise. just waiting. They're gonna give us some new waypoints, but we're just heading along, continuing on this transect line that we've been doing along a ridge here. No, pretty much all the urchins we've seen are alive, are living. So usually if they're dead, they're you get to see their skeleton usually, and it's white. And they wouldn't have their spines intact anymore. So yeah, all of these are living. So this one, for example, that we're coming up on is kind of a red.
What is that thing we've just zoomed in on?
It's another worm. They collected one the other day. I don't know what type of worm it is. We've been seeing quite a few of them. Yeah, so I mean, on this current dive, we're seeing I think we're seeing at least
There's
3
a
different
live sea
species of
urchins over there.
urchins. Yeah, so like this is some sort of Sedaris urchin, or a similar genus. So it's kind of got this red main body, and then it's got these thicker spines. And so yeah, that's living. It happens to have sponges. We've seen it several times now with sponges and ophiurites living on it. So there must be some sort of symbiotic relationship there. I don't know how beneficial it is to the urchin, but it seems to work out okay. And we've seen kind of those dark brown to blackish ones that kind of look like domes. Their spines are considerably shorter. So that's another group. Or I'm not sure exactly which one. It reminds us of these like spiky potato urchin that we see up on the Salazar Gomez Ridge. So it might be in the same genus or a similar genus. And then we've also been seeing these small kind of pinkish ones that are a little more flattened discs with also fairly small spines, and they're decorating themselves with pieces of shell and stuff, so hiding underneath them. So now we've seen at least those 3 today. Yes, at the other site we were seeing these ones, and maybe we saw them earlier today too, but I can't remember, that are also irregular urchins that have relatively short spines on it, but they're kind of elongated. They kind of look almost like a taco shape to me. Seeing a lot of these tube anemones, both early on in the dive and largely throughout. A number of tube worms we've been seeing. Some crinoids.
So these tube worm things, are they like always this abundant in other areas, or is it just in Antarctica?
I haven't seen these personally before, so I don't know if this is an Antarctica thing or maybe slightly beyond this region, but I'm not used to seeing tubes this large or this abundant for the tube worms. For the tube anemones, again, these are different species than what I've seen before, but I've seen areas where they're incredibly dense, much more dense than we've been seeing these tube anemones.
Yeah, because we saw quite a few of them on our last shift last time we did a dive, didn't we, as well?
Yeah, like it seems like all along this dive, I don't remember them being quite this abundant on the other dives and the other sites. Like we've been seeing them, but they seem more abundant at this location. We've seen some sponges too. a number of different sponges. Every once in a while I see a bright yellow sponge, which is kind of nice.
Something on the left-hand side that Maritza was interested in.
But who was?
Maritza.
Oh, it's like a sponge over there, yeah. That's what made me think of sponging. But we can move over and look while we're... I think I'm pretty sure it was a sponge. Yeah, right there. Yeah, that's a fish. The white, bubbly, cone-looking thing, I think, is a sponge. This kind of reminds me of, like, I'm probably getting the sponge group wrong, but... it's a sediment cloud. Okay.
That's one up the beds.
That's all... I thought it was a sponge, or that was like one of those bubbly sponges. It was just a sediment cloud from someone is clearing their tube. Yeah, it's a fish. Yeah.
It's a tripod fish? Yes. No? No. Yes.
Oh. Professor Tom,
Tom?
we need your urgent assistance, please, on fish identification.
Oh. But it looks like, no? But it's similar. I mean, the layer.
Are we down to only one laser again? Or we're just zoomed in? Oh, it's blocked. The fish core is blocking it. Okay. Yep. Thank you. Huh. Fascinating. That's alright. You can take the lasers off now that we have it and get a glamour shot. You know, I guess during our ascents and descents, there's been clearly a lot of phytoplankton and a lot of productivity, but we're not always seeing a lot of larger pelagic organisms in the water column. So that, I think, has been a little surprising to me personally. With all that productivity, I guess I think there would be more. We saw a decent number of things today, I guess, but yeah, not as much as I would expect for this much productivity. Yep, we're good, thank you.
And a party of night shift people in here tonight.
I'm caught in the loop of not being able to sleep.
Huh?
That's what it is.
It's the anticipation to get your sediment cores from RAV.
Yep. Are we going down slope now? A little bit. You can see the lasers are up higher. Yeah, so we've kind of hit the summit of this local peak that we are working up, and we're heading back down. Another Dumbo octopus.
This one is
Yep.
Yeah. Something's been digging, and there's another octopus hanging out in there. Or it dug it, I don't know. Probably. Looks pretty fresh too. It's not. Can we look at whatever this is over there? Hmm. I think they're worms out. Alright, thank you. Go ahead and keep moving along. See what we see. Like subtle, I feel like there's subtle changes in the community. Right here across this ridge that we've been going over. At least we've been seeing some organisms we haven't seen before. You know, something I thought was really interesting today is we saw this one area, there was a lot of benthic jellies, so that was really interesting. Like dozens of them.
So the sediment can be different depths in different locations. Some areas, the rock is literally sticking out, so there is no sediment, which is what I was saying earlier. With gravity core, we have to be kind of careful and try and choose an area where there's big pockets, so essentially a few meters of sediment that we can actually stick that core into. This area right now, I don't know, but we've mapped all of this area before doing the ROV dive, so we could check how deep the sediment is. And occasionally when we do pass over rocks and walls, so there's obviously no sediment there, and that's just exposed bedrock from underneath.
And all these things we see sticking out of the sediment are tubes from tube worms. So most of them are now vacant and just left behind tubes, but there's some still have residents in them.
I've got my coffee now, I'm happy. Did quite a smart thing this evening before my roommate went to sleep. I've pre-made an espresso so that I could be loud with the espresso machine in the room before she went to sleep, because I knew the long ROV dive. It's here. I put it in the cup so it's cold, but it's ready for nighttime ROV dive shift. Yes, if you look up some scientific papers, or maybe if you try and Google CT scans of sediment cores, marine sediment cores, you'll see. So what we do is essentially the CT is kind of like black and white, a bit like, you know, an X-ray, if you like. Sometimes we used to in the past do X-ray, but now technology is so good, CT scan is better because it like layers through the sediments. So you don't just get the top bit, but you get really nice layers, and that you can see stuff like if the sediment is mixed up, or if there's rocks, or coarser grains, it comes out as different colors. Sometimes you see lamination, so perfectly layered kind of sediment, which is very satisfying. And what we do is we stick it in some computer software and basically make it all different colors to emphasize the different kind of fascies, we call them. So we can like split the types that we see in the sediment, and from that some people deduce, for example, how fast these ice sheets are retreated, because different kind of sedimentology properties and course and size of grains lets you find out things about the retreat of glaciers and the kind of conditions, whether it was a sub-ice shelf, so if the ice shelf was overhanging the area, or whether it was alternatively an open marine kind of setting, so you just had sediment falling down essentially from the top, like organic matter, and sliding down the wall. but this amount of sediment we get is very different in different places. So some areas like Antarctica, because it recently was covered by ice, there was kind of no sediment build up underneath. When you then get loads of... Oh, excellent, we're on the move. So yes, if the area was covered by ice, essentially you kind of have rocks sticking out and you don't have much sediment, whether in open ocean environments, especially where there's continental shelves. Oh.
You're like, uh-oh.
But yeah, so where you have like sediment areas, for example, off the Portuguese margins, something called the Iberian margin sediments, very productive, and recently cores of over a kilometre of depth have been drilled, so it's a kilometre of sediment especially, but you need kind of very special ships with drilling facilities that can just keep drilling and drilling as you keep down on these chunks, metre and a half. And the way it works is quite interesting on the cruisers. Basically, people shout core on deck and the whole team, the core kind of passes through the whole team, so people cut it in half. Other people describe this colour with the little book that we were telling you about earlier. Then it gets passed on. People try and touch it and smell it and feel it and see how big the grains are in the core. Then it gets passed on further and people do some magnetic analysis and then they wrap it up really nicely. You have to package them quite tight because otherwise they dry out and they become really hard to sample. You can also grow mold in them, so we tend to keep them in the fridge.
Alright, yeah, they just gave us a new waypoint, so we're gonna make a U-turn. We gotta wait, we are gonna have the ship move before we can make the turn, so because the ship's in our way. It's on the wrong side. We're on the opposite side of the ship from the direction we have to make the turn. So just bear with us for a moment as the pilots get the ship to change position so that the ROV can continue flying.
Underway.
RV coming.
I wish I could remember what he just said, because that was really funny. He just said like a whole new world of waveforms or something. Yeah. Yeah, we're still going downhill, but we're just going downhill in a different direction.
But yeah, other interesting things we can do with the sediment is I've been extracting this pore water, and this essentially looks a bit like a torture experiment on the laboratory tabletop. So what I do is I've made some holes in these core liners before. I actually got my brother to drill me these holes in these plastic core liners, so just in the little push cores that we collect with the ROV. And then I get my sediment and that subsample it from the ROV sort of wider core liner thing and stick my tube in it with holes. I take them up so that it doesn't leak out because the sediment is fairly liquid actually around here. Then we bring it up, we put it on the table, and I stick these kind of like thin long membrane things into the sediment attached with a syringe at the end. So essentially what you see if you come up to this thing is a bit like some medieval torture device. There's just like syringes hanging out of the sediment. But if you open the syringe plunger, so you create kind of like negative pressure and it sucks out the pore water out of the core. And it's quite interesting because then you can analyse this the chemistry of the bore water, which I'll do when I'm back at home in Cambridge, and compare the chemistry of the spore water to the chemistry of the organisms, these fossils we find in the sediment. And from this we can better improve our understanding of sort of how the various chemical ratios in this calcium carbonate foraminiferous shell, so these organisms, how it relates to different environmental parameters. For example, like the amount of past oxygen that was present when the shell lived, or the amount of past carbonate ion, or dissolved inorganic carbon, so different various things like that. We can also measure something called isotopes, and the ratios tell us which water masses were present. And now this is very important because one of the main mechanisms we think that these ice shelves have melted is this warm water came in under the ice shelf and still does, and due to anthropogenic reasons, so human-induced climate change, and essentially melts those ice shelves from underneath. So yes, what we do want to know when this warm water appeared on the shelf and how fast that ice sheet retreated. Well, maybe. Thank you for the comments about sediment torture. Indeed, there's been quite a few people on the ship taking photos with my sediment torture devices. And what's exactly. Little do you know what geologists like to do with sediment. And the comparison is quite funny. The biologists are only really interested in the top sort of 5 centimeters of the push core sediments that we collect here. So what tends to happen is they sample these 5 centimeters and then chuck the rest of sediment back overboard. Sometimes one of the geologists quickly runs along and goes, "Please give me your sediment. I love sediment. I want more sediment." And then eventually, you know, you get a reputation for liking sediment, so people start to donate you the sediment. Personally, I really love ocean sediment, especially from Antarctica. It's so nice. I get so much sediment from this cruise already. I'm really happy with all the amount of sediment. It's very exciting. I think even Anna has had enough of the sediment stories.
I love it.
I love it, but
please
shush.
Sediment plus something.
We are falling asleep with the sediments.
Maybe I'm a good bedtime story. I hope you're still awake on the live translation online.
When I have some insomnia, we'll call you.
Yeah, so dating methods for older sediments. I do indeed use radiocarbon datings of carbon-14. For more recent sediments, I'm sorry everyone in the room, I hope you're still with me. For more recent sediments, I often use something they're called lead-210, so it's a similar type radioactive kind of isotopes, but the half-life is very short, so we can only use that for the last 100 years or so. Radiocarbon dating I think is very interesting. I have a lab in Cambridge for radiocarbon dating, and it's quite interesting. It basically looks like I'm a plumber when I show people what I do in the radiocarbon lab, so it's this vacuum kind of gas line. Because our samples, we turn them into gas to get them into the sting, and we have to try and get them into an individual reactor around this sort of circular pipework. Oh, what is this? Maybe it's more interesting than the sediment story. But with the pipework, essentially you want to kind of isolate your sample into a particular reactor. And if you imagine gas is sort of very flowy, like it's hard to just put it in a container, so we have a lot of valves, and it looks literally like kind of a plumber thing with loads of different valves. I twist for a few hours. Once I get all my gases into the reactors, I have 11 reactors that I can run at the same time. I then turn the reactors on, and it brings the temperature up to around 1,000 degrees. And this causes, and we also have iron catalyst, actually, I forgot to say. And this causes this gas, this is CO2, to turn into graphite. So essentially then the reactor spits me out a graphite cathode. So literally graphite that we then give to a different lab. We have some collaborator laboratories. You just stick it in a certain mass spectrometer and we get the date of how old something was.
There's a different type of urchin we haven't seen. Looks kind of like Hydrosidaries or something like
yes.
that.
Is there any specific events or
We can go ahead and keep going. If we see another one, we can look at it. We got time waiting.
For me?
Okay. Yeah, we can go to stop and look. And
I don't know. Well, I'm a geochemist, so I'm not necessarily like your classic rock geologist. So I do lots of chemistry in the lab, and I've always liked chemistry. And what I like about like geochemistry and geology, kind of our discipline, climate change, is that it's very kind of multidisciplinary. So I get to think about physics, occasionally biology, which I find very scary. Sometimes mathematics. I do some computer programming, and a lot of chemistry. So there's never a boring day. You're always learning something new. So I think it's really cool how basically it's not very like narrow in one discipline. It's very kind of multidisciplinary thing. So
yeah, that's a pretty one. This might.
it's not. I said it's scary. I didn't say it was boring right now.
I don't know. It's scary.
Cool, thank you. There is a question up there about what are some of your favorite deep sea organisms. So for me, right now I've been working a lot with corals. I actually have students in my lab working with meiofauna as well, so we do some sediment stuff as well in my lab. But for me, I really like the diverse communities, and I particularly like the mesophotic ecosystems. They're really colorful with really diverse communities that change quite rapidly with depth. And so I find them quite beautiful and amazing, and just you're always finding something new. And so more about the really interesting communities of organisms than about any one particular organism. Although octocorals are, I like working with them right now because it's like solving a puzzle, because as we do genetic analysis, we're finding that our understanding of their evolutionary history and relationships are really quite different than what we had thought. And so it's making us rethink the morphological data that we're using. And so right now I find that really intriguing. But, before I worked with harpacticoid copepods, so I also find them cute, and working with them was also a challenge and a puzzle that I enjoy. Anna? What's your favorite organisms?
Actually, my favorite is Ctenophora. I really like it, even if I couldn't never work with. And I actually work with corals, but from the ecological and marine ecology perspective. But it's true that I don't know. I was thinking actually that Ctenophora maybe will be the next step, but don't know. Let's see.
Maritza?
Don't say sea
Uy,
pen.
está, sí, organismo favorito.
Sea pen, you can't say sea pen.
I don't know. My favorites. The sea pen and sea star. I love sea star. It's so cool. Wow.
Oh, wow.
No.
That's some nice fat sea cucumbers.
Yeah.
And look at.
Teresa, do you think
And there's one of those big polychaetes.
Professor Sea Cucumber, please could we collect one of these? I really want to I want you to dissect the sea cucumber.
Mine. Fish ruined it.
These Aphrodites. Do you think it's possible if we collect one?
Erin?
Sure. Why not?
Yeah?
I love it.
That would be really nice. Thank you.
Yeah, can we put it in with the other?
Yeah.
Okay.
Yeah, because they are more or less the same depth. It's okay.
Okay.
The last.
Because, yeah, we don't have a lot of space, so.
Okay.
I think this is a really
We'll put
juicy
it
sea
in 1D.
cucumber. Yes, I think they can hear you actually.
Stack it with the other.
Yeah.
The holothurian, the sea cucumber, the big
Olá, Kelly. Então, prima, está tudo bem? Nós estamos aqui na Antártida, estamos a fazer uma expedição científica pela primeira vez. Já chegamos aqui há algum tempo e estamos a estudar, somos uma equipa multidisciplinar de todas, temos aqui biólogos, temos geólogos, temos oceanógrafos e vamos estar aqui durante algum tempo. Sempre que quiseres assistir, és muito bem-vinda e sempre que quiseres fazer perguntas, nós tentamos o nosso melhor para te responder. Nós não vamos interagir com os pinguins, quem me dera, porque neste momento o objetivo principal do nosso trabalho é estudar o mar profundo. Está bem? Obrigada pelo interesse. Beijinhos e fica bem.
Muy bien.
So we're just gonna stop here and we're gonna collect the sea cucumber, and then after I'm gonna try to figure out where we can suction up this polychaete.
What is this thing on the left of the sea cucumber?
It's a polychaete.
A sea mouse.
Oh, sea mouse.
Yeah.
I thought polychaete was worm.
It is.
That you are right.
But it's a different.
Oh.
And then
We have a lot of them in the North Sea,
Mm-hmm.
and it's really cool to see them here. I already dissect one for a very.
Oh, very nice. Can you, next dissection of this large guy, can you put my GoPro on and do a time lapse of the dissection? I think it'd be really nice.
Okay, give me.
Perfect. Oh, look. Oh, its belly is quite nice. It's sort of a funny texture.
Do you want to dissect?
I'd love to dissect it, but I have to do my sediment course. But maybe if you collect one extra, I can just, if you leave it in the bucket, I can come with Rachel's hairdresser scissors that I used for my haircut today. That would be perfect. I would like to say to Dr. Professor Teresa's cousin, who I believe might be listening to us in the audience, Professor of Sea Cucumber. This is the sea cucumber for Teresa. It's very juicy, and Teresa is very happy. She's got a very big smile on her face right now at how nice this cucumber is.
one being.
It is so good.
Sure.
Yes, please.
Oh, I need to lock this.
It's beautiful.
Yeah.
Which location is this going into, Erin?
1B, one boy.
BB. One A, B.
Kelly, nós neste momento estamos aos 680 metros de profundidade e nós vamos estar aqui 6 semanas. Obviamente que sim,
Awesome, thank
esperamos encontrar novas espécies. É esse
No,
o
we're
nosso
gonna
trabalho.
Suction that polychaete.
Teresa.
I have no clue which one to put it in. Yeah. No, then I'll get them confused. There's a fish in there. We could put it in. Did the fish from 7 end up in 6, or is there a fish from 7 too? 2.
6 was
The bonus fish. Let's put it in. Oh gosh, there's a gastropod in 8, so let's put it in 8. I think we could put it with a snail.
Are the sea cucumbers soft and squishy, like a nice?
This one is hard.
This one is hard? Or is it like quite solidified? Quite like how soft? So a bit like a fish kind of
Do you think?
texture, maybe.
Oh no, we can put it.
Softer?
We can put it in, yeah, just very-
Harder than fish, so like very
you
kind
have
of
polychaete, yeah, that-
spiky, like a nice dinosaur, and soft like a kitten's tummy on the bottom. On
But
the
you're saying fish is gonna be gone out of this.
Professor of sea cucumber.
No, we're
cannot
gonna put
wait
it in
to
8.
see the juicy
6
guy.
has like a bonus fish that wasn't intentionally caught apparently.
Hopscotch over.
What do you have in 1?
Oh, I need to.
Sediment
I think we can index to 1. I think what's in 1 won't swim away.
Alright. Sure.
Otherwise, in 7 there is this snail. This
No,
animal.
7 has a fish.
I have 8 fish.
No, I have 8 as a snail.
Okay,
I hope 8. I'm pretty sure 7 has a fish in it. There says fish in 7, so I think that one's wrong.
Yeah, 7 has fish in it.
Yeah.
Okay, so we have
Eight has something else.
Put it in 8. Yeah, I don't want to put it where the fish is. I think that might make for a. Yeah. Yeah, 8 has a gastropod. And then we can index to 1.
For waking up. What is that thing on the left? Maritza, ¿qué es esta?
It's the same.
It's a smaller
Out it.
I
polychaete.
mean, it's
Oh, look at it go, so smooth when it goes into the sea hoover.
It's not a sea cucumber, it's a polychaete.
I know, but it's just that goes in so nice.
There it went. It's in there. Can we hop over and grab the second one over there too?
I'm putting him anywhere.
And 8 will add them. They can go in the same place because we're collecting at the same time.
Which one?
On the rock, there is a little one. Thanks.
Pop over some.
Yes, it's an 8. And we're gonna grab a second one and add it to 8 because there's a baby one.
okay.
Make America really happy. Actually, it's not on a rock,
I think
it's
we
on an
have
urchin,
people
I think.
interested in how the sea cucumbers taste again online in the chat. So I believe Professor Teresa told us earlier that she has tasted the sea cucumber and it was not very
And you
good,
look
so
for
she
one?
does not recommend it.
¿Ni lucho?
What? Is it something?
There he goes. Alright. Watch.
Okay,
Alright, there you go. Thank you. And yeah, we can index the one.
Eight, no. Es
Saki?
yeah,
Saki number 8?
We can start the section. Yeah, we're good.
And ¿qué esta?
Polycake.
Muchas gracias.
Yeah, so we just sectioned up 2 polychaetes. Apparently this group of polychaetes, it's hard to tell them apart unless you have something in your hand. So it's like cryptic diversity, so that means like 2 species can look the same, but genetically, for example, they're different. Or there's really small differences that you can't see. So we're collecting, these are one of our target organisms on this expedition to try and look at the diversity that we find. Onward we go. See if we can make it to this waypoint. U-turn. Downhill we go.
So we were hoovering up some of these beautiful sea mice. So we got 2 specimens. I think people are wondering in the chat.
Oh yeah, I already.
Oh, you explained already?
Yeah, I did. Thank you.
Someone online says they quite like the taste of the sea cucumber.
I would not.
Oh no, this was
Yeah.
jellyfish.
Yeah. I don't think I'd want. Yeah, I don't know. Sea cucumbers are. There's a few different. There's like the heart, like that last one's gonna come up and I guess maybe. But the other ones are gonna be this gelatinous like goo, and then there's others that come up that like get bloated on the way up, filled with water. You have to like pop them. So it's like a little fountain of water just running out. Yeah, they're interesting.
I think the round one was also a sea mouse. So what are we doing now, Erin? Are we flying to our next waypoint? Is that right?
Yeah, we're heading to our next waypoint. We made it to, yeah. In the round one, you might be talking about the urchin that the smaller polychaete or sea mouse was resting on, so we left behind kind of the roundish urchin. But yeah, we got new waypoints because we hit the end of our planned dive, and they gave us new waypoints. We're kind of making like a box back towards where we were earlier, so we had to make a left turn. The ship is now, it took us a while to do it, and I think it helped when we did the sampling, so the ship had time to move out of our way because the ship was actually like practically right over the waypoint we needed to head to. So we had to wait for the waypoint, for the ship to move away from the waypoint. And then we're going to be making another left turn here when we get to the next waypoint and heading back kind of parallel to the transect that we were just doing. And again, they're going to have to keep moving the ship because the ship's on our port side, right? My brain's working right this early in the morning. And so we have to, the ship's like basically in our way all the time right now, so we have to follow the ship. The ship's gonna have to move out of our way as we're making these positions to track the new waypoints. So we're just doing that, so we might have a few pauses as the ship does that. Although it's gotten far enough ahead of us now, maybe it won't be an issue anymore. But we're almost to waypoint 15, and we'll be making another left turn. So yeah, we're still seeing pretty similar community to what we've been seeing for the last hour or so. Some sea cucumbers, some urchins, some fish, these polychaete tubes. Yeah.
Professor Teresa, there's commentaries for you in the chat.
Is it? And left turn, here we come. And we're still kind of going downhill, so if you're wondering why like the lasers are so far ahead, it's just we're going down slope, and
Oh,
we're
look
also
at that. Is that the snailfish
further
thing?
away.
New waypoint.
I don't
So
know.
it was just an
RV brakes, new waypoint.
Oh, what is that? What is that?
The
¿Qué es esta,
anemones
Maritza?
and crinoids, and then I think that's some sort of like polychaete. Tube anemone. Yeah. No, we can keep going. Looks like. Yeah, sea cucumbers definitely have different textures, I would say. Some are a little harder, some are a little softer, some are a little more gelatinous.
I think so. This area here, we're in a slightly shallower bit, and we seem to have a lot of very nice soft sediment. So there's probably some nice sedimentation going on here. Might be quite a nice thick layer. I think we're planning to come back on a dive a bit later and sample the sediment here and compare it to where we started off at the beginning of this dive. We got some sediment from the sort of deeper sides. So we'll see what the difference is.
Yeah, so I guess we're nearing the end of this dive. Let's see, what's our estimated time to get back on deck? What, 5 o'clock hard on deck, so our off bottom time is what?
Oh, Professor Thomas finally
what?
joined the room.
4:15? Yeah, between 4:15 and 4. Oh, there's a nice sponge. So yeah, passing another one of those polychaetes. So yeah, between 4:15 and 4:30. So in the next 20 to 15 minutes, we're gonna have to come off bottom. So yeah, we're just.
And I think for those in the chat that were here earlier for all the fish questions, we've got a science crew swap in about 5 minutes' time, and Professor.
Yeah, so yeah, in the next couple of minutes, yeah, our science team is changing out, so you'll hear a little more rustling and noise in the background as the next shift comes in, and then we'll start swapping out positions. And also as well, the ROV pilots are changing out. For the last 15 to 30 minutes of the dive. And they'll be coming off bottom, but what we're doing for the rest of the day is we'll come off bottom in the next 15 to 30 minutes, and then we'll be processing our samples. And then while we're doing that, the ROVs, they're going to be conducting a gravity core that Schmidt has been talking about. And they'll also change over the ROV, process it, and get it ready for the next dive. And we'll do a short dive, only about probably scheduled scheduled right now for 4 hours to do the push course, but it might not be much. It might not even be that long. I guess the push course is usually taken quicker. So we just have Jess and Claude coming in, and Andy and Sunny heading out. Yeah, I see it's buffering, so yeah, don't worry, I didn't really introduce anyone, just kind of recapping what we're going to be doing coming up and what we did. And yeah, the next team is coming in, so I will let them take over. I think they're all in the room now, so I will see you all at the next dive, probably. Have a good night. Everything's pretty much full. If you want to stack things, if you're going to collect anything, there's only 15 to 30 minutes for our coming left. The only thing that's still open is a few coral pigeons, but we have only seen sediment in the last few hours. So. But if you see corals, just collect them.
Okay.
So that's even, yeah.
We're able to take a sample?
Those have all been taken, like everything has been taken. If you look at your waypoints, they're going backwards towards the ship. So it might be some occasional pauses as the ship has to, it's like
The backpedal.
Yeah, exactly.
So other than anything else, you call
Yeah, maybe coral and if you see any, but I'm not super cool at this point. But yeah, this came on at the end, I think, super coming up.
So, pretty thorough.
Yep, awesome, thanks Tom.
See you later.
See you later.
Hello to everyone in chat. We've just done our shift change, so we'll just do a quick introduction so you know who you're listening to. I'm Tom Lindley. I'm a fish curator at the Museum of New Zealand Te Papa Tongarewa, and you will have, well, if you've been listening for a while, you would have heard Sveta bullying me for the last hour or so, throwing fish questions my way while I'm trying to have breakfast. I'm also joined with Pedro.
Yes, hello, here's Pedro Martinez. I'm a marine biologist from Spain, but I'm working in Germany at the Senckenberg Institute, and I'm happy to be here in the Antarctica.
It's snowing outside. It's beautiful.
Snowing also here inside.
Hey, there we go.
I mean, in the deep sea. As you can see, all these particles flashing down to the deep sea, coming from the water column. We call them marine snow. So it's snowing outside and also in the water.
Excellent segue. So we're just approaching the end of the dive. We've maybe got 20 minutes, half an hour left of bottle time, so that we can make it up to the surface in time for, well, when we've decided to wake everyone up. And we have a pretty full sample tray. So unless we see something absolutely spectacular that we can cram on top of something else, we'll probably just be doing a visual transect, seeing if we spot anything cool for the last few minutes of this dive.
We're now at about 700 meters. We started the dive yesterday at about 1,000 meters. Here we see that there is a lot of fauna. Also the ground is not so even, so we see that there are a lot of small mounds. These are mounds from worms. We see that there is a lot of bioturbation. We see here some holothurian. These ones, we have seen them also in the deep, and they are quite common here in the area. probably belonging to the genus Scotoplanes.
Are they ones we'd casually call sea pigs? For people like me who don't know holothurians. I think they come under that heading.
Outside is very clear this morning. It is a little bit of snow, but we don't see a lot of icebergs, so everything is good.
I would say come more to the west, yeah.
Early.
We see at the bottom also some traces. It is for us very interesting to know which organisms are leaving these traces at the bottom, because then we can also make an interpretation of which organisms are living there, even if we don't see the animal. And also these traces are known from the fossil records. So we can make a better understanding on how was the habitat in the past, with kind of organisms were leaving these traces, even if we don't have the organism itself.
The trace fossils are fascinating. I did some work with them recently for the first time, and the body fossils that we usually associate with fossils, you know, the skeletal remains and the outline of the animal, it's incredibly detailed, but it is a scene of a dead body, if that makes sense. It is a dead organism, and it has this sort of sense of death around it. But the life traces, those they feel like a snapshot in time. It's really, really interesting. So I was looking at some early tetrapods, so before we even have amphibians, like the the fish that walked up onto the land essentially, and it was this scene of a muddy bank, sort of like mud skippers, and you could see the shapes of the raindrops that had hit the mud, and then these sort of fins but with little claws pulling themselves up along the mud. And it was weird, it was just this snapshot of time. It just felt so alive, and I'd always seen the sort of body fossils, which are always a dead body, and this was just such a snapshot of life, and I found myself sort of like rooting for this little proto-terrestrial vertebrate pulling itself out of the ocean, and what a significant moment that was. And so yeah, I got into the traces. They tell a story.
There are of course some traces that we don't know which organisms are doing. So now we have a lot of video and photographs from the seafloor, and we are improving our understanding of who is leaving these traces. Yeah, I also saw this tripod fish. Do you saw it, Tom?
It's a genecthlid pretending to be a tripod fish.
Okay.
I've got a similar sort of convergent evolution going on, but they're doing the same thing. They're a predator that's lifting themselves up off the seabed, so they can better detect prey. And there's two main groups within the genecthlids. There's ones with sort of little rounded pelvic fins, and they're the ones that sit right down on the bottom. And then there's these ones with the stirrups. But I've never seen one with such huge pelvic fins. I've not worked with the group very much, but that was a surprise when I saw that come on the screen. That was an amazing animal. Luckily for them, the bioboxes are all full.
You think that this might be a new species?
It might just be one I'm not familiar with. The group's kind of new to me, but they're really growing on me after this trip, so I might have to dabble. Some of my ideas have been backed up by a couple of experts who've been watching on the chat and sort of messaging me as well, so I'm very grateful for those. I'm just the mouthpiece to smarter people.
How it is the fish fauna here in Antarctica? Is this something that is unique, or many fish species are living only here, or it is more connected to other oceans?
There's a massive radiation just here. The notothenoids are the main one, the sort of ice fishes, and I don't think I made that common name. Doesn't encompass them. Anyway, there are multiple groups here that have sort of radiated out, and they're just super specialists to Antarctica, including really interesting things like the ones that are lacking any blood pigment. So they just suspend oxygen in their fluids of their blood, so they have clear or white blood, and that's pretty unique to down here, and it'll probably only work in these cold temperatures and well oxygenated water.
Okay, this is because there is so much oxygen in the water that they don't need the hemoglobin.
It seems to be part of the cold tolerance as well. They have natural antifreezes in their blood as well. So it seems to be part of maintaining that balance so that they can go so cold.
Here we go, we had some small disturbance.
No worries. What time do you think we'll have to leave the bottom to be up for the final? Yeah. And just enjoy our walk. We're having a chat in the mess just before coming on shift. When we come up off the bottom a little bit, you'll see some of these, what look like transparent little fish swimming along. And those are actually arrow worms. But I've trained the visual centers of my brain to just respond to fish too much, so I keep seeing fish where there aren't any. And those are ones that really make that itch. Fish, fish, collect. No. Worm pretending to be a fish.
Yeah, these arrow worms are very, very big. Normally in other oceans they are smaller. But in the polar regions, the plankton also tends to be a little bit larger. Oh, what is that? Reddish one. We have seen also some very beautiful sponges. Now we see a Ascidian, a very big one. Tunicate.
Is that what that was? The tower-like one?
Yes, yes.
I hadn't seen the stalked ones before, these ones that lift themselves way up. That was a new one on me.
The white one, it looks like a crinoid. A lot of tubes from worms.
Yeah.
Some sea star. Sea pigs, Holothurian fish.
This little area here looks quite turned over. There's obviously a lot of organisms.
Yes, yes. But you also see that there's a lot of bioturbation. You see that. So there's lots of activity in this area, which is a little bit more elevated. In general, we have here a lot of filter feeders, so they like these elevated areas where the currents are a little bit stronger and they can get more food. Here you see a sea urchin and the trace left by the sea urchin. So this is the kind of pictures that we need in order to assign the traces to the organisms doing it.
So for now we're conducting transects. We've got our laser scale on, so you might just make out that couple of green pixels right in the center of the image. Those are 10 centimeters apart, and we can trace back the ROV speed, work out what area we've covered, and then we can actually get quantitative data as well. So we'll get the density of these animals, and we can plot the habitat transitions. While we are chatting away and trying to navigate things, there's multiple other members of the team who are making notes with timestamps of all these transitions. Oh, there's one of the arrow worms there, just swimming, trying to be a fish. So yep, there's people constantly logging the animals we're seeing, and sometimes we see quite an abrupt change in habitat. and we notice it. But sometimes it's actually, once you look back at the data, you realise there's been a shift. Sometimes it's so subtle. We're probably only seeing, maybe 5 meters or so, and it can sneak up on you. You can not notice that there's a change until it's happened.
Again, one of these tunicates, and Holothurian at the right side. Now we pass it. I think they are called Molgula, something like this.
The little plunderfish down there. I'm seeing a few of those. I'm used to the chunkier sort of Pogonophrine ones, which are more like a monkfish almost. But these are quite pretty little ones, but they have a little lure on their bottom lip that they use to hunt, or at least we think they use to hunt. We're not entirely sure. This little fleshy outcrop, and it seems that they accidentally bite it themselves every now and then. So like we bite our tongue when we're eating, they accidentally bite their own lure sometimes, so they can look a little bit raggy.
Some jellyfish in the water also. Oh, look at this. Anemones.
About 12 hours ago now, because this has been a long shift, but we disturbed a jellyfish meeting on our last shift. It's just suddenly just this whole patch of them.
Oh. A very beautiful anemone now.
Yes, that's our final task. I think we've only got one left.
We have seen also this anemone lying at the side and rolling, you see that? We have seen them in different parts of the Pacific Ocean also. We don't know if this is really a strategy to roll, to disperse a little bit. We call them the rolling anemone.
And you think it might be to move to a new site?
Yeah, maybe yeah.
There's some swimming ones in New Zealand that travel around, and some of them do tend to walk along on their foot, and they battle as well. I didn't know that. A friend of mine did that for his master's thesis, and had these incredible time-lapse sped-up videos of these two sea urchins just whacking each other. It was amazing.
That's great.
The rest of the vessel is starting to wake up. We've bumped into a few bleary-eyed people starting to wake up in anticipation of the ROV coming on. We're working at a slightly different schedule right now, so it's all right for us night shifters, but a lot of folk are having to get up very early for the frenzy of the sampling processing. I've got a challenging bit of sampling to do when these samples get back on deck, because a colleague of mine asked me to collect some ice fish blood samples, because they have that very special blood we were talking about earlier. So I've got to figure out how to do that. Yes, if we're ready to go. Habitat hasn't changed, so we can do it to our last thing. One of our final sampling tasks is to just take a Niskin bottle for environmental DNA sampling, and this will be the sort of bookmark for the end of our dive. So just along the left side of the ROV, there's a suite of bottles that are open on both sides until you fire a link and the bottle closes. So as we're moving along, water's constantly passing through these and flushing them through. And then when we decide we want that particular bit of water, we can snap closed both ends. And number 6 is the only one left.
ROV bridge, do you want me to keep moving to the south?
Bridge ROV negative. We are just gonna get ready for recovery now.
Understood.
Thank you.
So we're gonna start the ascent.
I think we can.
Yes, correct.
Bridge ROV.
ROV bridge.
So with our final task complete and pretty much every sampling device on the ROV filled, this mammoth dive is now complete. So we're going to start heading towards the surface, which I think is when we lose our viewers. So for anyone who's stayed up with us, and I know a lot of people have watched a lot of this dive stream, thanks for the company and thanks for the questions. It's always good to answer a few questions when we're in a quiet moment. So that's a thank you from me.
Yes, and also thank you very much. This was a short dive for us, but in total it was a very long dive. So we are very thankful to the ROV pilots that they did a very great job collecting all the samples.
And putting up with our demands, unreasonable requests.